Hello, everybody, and welcome to the show. We are going to be here with Warren Booth and Dave. And first things first, I'm going to give a big shout out to USARK fighting for your right and my right, as well as everybody else in the community's right to keep and breed the reptiles, amphibians, and just actually crosses over into other species too, because the same legislation that is prompted to go after reptiles is often going after other exotic animals. And USARK, mitigates a lot of these, this real poor legislation that is targeted at the reptile hobby and the exotic community. And so please support USARK, Phil, Daniel, Vanessa. They all do a great job. There's many other people, the lawyers. They've been growing in strength, and USARK needs our support to continue to grow in strength and to be able to allow us, what we like to do. Whether you're a hobbyist, keeper, whatever, you can support them any way. Could be a member, buy merch, see them at shows, spread awareness. Go to the website, see what if there's any current legislation being proposed. So please always support USARK. So we're going to bring Dave, and we got Warren Booth, and the topic today is going to be genetics because we have Warren Booth here, who is a geneticist and has written many papers on parthogenesis, I'm sure, as well as many other things related to genetics. And so probably kick it off with Dave. You want to say something? I know you and Warren actually have a little bit of history. You guys seem to be friends for a little while.
Yeah, for a little while, not so much anymore, but we had a pretty good run back in the day. On a somber note, so we lost a couple people in the hobby this past week. I just want to send a shout out to Ryan Goodman from R&B Reptiles, guy who got me into podcasting with Benjamin. He passed away over the weekend. Very tragic, very unfortunate. And Ryan Galash out of the St. Louis area, another great guy. Talked to him all the time at the shows. Younger guy, I think he was 41, also passed away over the weekend. So hate to start out on a sad note, but honestly, just kind of want to throw a shout out. Sucks.
Yeah, of course. That's terrible, man. I didn't know that. That's two people from the community at once. That's not good.
That's rough. But yeah, just a shout out to them. But yeah, I love Warren. Love talking to Warren. I think this is probably our fifth or sixth time we've done this, but this is the first time three of us have been together, right?
Yeah. I've met Warren briefly at Tinley. I talked to him about genetics for about like 10 minutes in the lobby. And then I talked to him more at the Southeast Expo. Bob Vooz, we talked a bit genetics and up at the rooftop there. And now this is the podcast. And actually, I told Warren, I was like, oh yeah, I wanted to have you on. And I mentioned you to Dave. Dave's like, he goes, man, I just did one like 3 days ago with him. It's like, or like just the last week. But then you were talking, Warren messaged me because of the topic we were talking about in the previous podcast. And so I think, well, this is like a good opportunity to discuss a particular topic that I think is not touched on a lot and is very interesting. And that's inbreeding in captivity as well in the wild. Like what is a safe level? Not so much because it's going to vary between species, but the interesting thing is like, there's so many ball pythons imported, and that's kind of what got that was kind of the topic there. It's like, well, there must be a lot of genetic diversity because you have ball pythons being collected from Benin, Ghana, Togo. I don't know if they ever get them from like Burkina Faso or like, any other neighboring countries. I do know when they go to look for new mutations, I'd always hear from like people that have collectors that they go to new locales to find these mutations, which makes sense because a lot of these mutations probably exist within these populations and reproduce and pass on those genetics. So anytime they're getting a new mutation, there should be some, very different genetics there. And so, Warren, you were talking about with me, you told me you educated me on that. Hey, we did some, I guess, did you sequence their genomes? What is the process of finding out how closely related the ball pythons were? And what was your, ratio of these animals that you were, studying?
Yeah, so we've looked at the genome of or genomic diversity of a bunch of different species in captivity. It's all related to our work on parthenogenesis. Whenever we sequence the parthenogens, we always sequence the mother and the father also, or the mother and the putative father if a male was present also. So it means we've got that background level of genetic diversity estimate right there as well. And of course, you see parthenogens have collapsed genetic diversity as we expect, but we always look at it in relation to the genetic diversity of the mother. In all of the ball pythons that we've sequenced, and it's been dozens of ball pythons over the years, boa imperator, several species of rattlesnake, king cobras, some other old world pit vipers, we find that genetic diversity is really low, but ball python genetic diversity is extremely low, as is boa imperator genetic diversity. Much lower than we would expect or predict from wild populations. And the way we look at it is that, whenever ball pythons, boas, and so on have been in the hobby for a long time, the hobby is largely driven by color and pattern morphs. And what's happening is people are buying pairs I believe they're buying pairs from the same litter or clutch and breeding those multiple times. They might be introducing new genes, but they're adding in genes that they've already got. So there's a level of background relatedness to them also. So it doesn't matter whether we go for short read sequencing or entire genomes, we find their genetic diversity is really lower than we would expect. And what I was thinking, and I messaged you a couple of days ago, was it would be really cool to look at genetic diversity within these captive farm ball pythons that are coming in.
Right.
We don't see them coming in the numbers they used to come in, but they're still coming in. But I don't know what regions they're coming in from. So we see the ones that come into Outback every year. I don't know if they're being shipped in from multiple different countries or only a single country, but it would be great to get 10 or 15 of those just randomly, just shed skins, so that we could then sequence their genome to look at what is present in the wild populations, assuming that they're collecting those ball pythons from a short radius from where they're being shipped from ultimately.
If you could get one of the ones where it's like, I know the bells and stuff, I mean, they used to get like 10,000 at a time. So that's from a large, farm operation. And I believe all those females too, I mean, they're catching them gravid. Yeah. They're letting them lay their eggs. And so they're having to go from a very large range. In order to get that many gravid females, they're definitely having to go on a pretty far range. Mm-hmm. Now, my question is, and I think this is an interesting one. You see the ball pythons, their allele, I guess, diversity was pretty similar. Like, it's not like you expected them to be have more diverse alleles from one another.
Yeah.
Do you think that certain species, kind of like cheetahs at one time were almost nearly extinct and they're bottlenecked, and so all their DNA is very, very similar? Now ball pythons have a pretty large range, so I don't think that would be the case. But do you think certain species, would naturally have a lower allele frequency difference?
Yeah, that's a really good question, and the answer is complex. So for people that don't know what genetic bottlenecking is, a really simple way to think about it would be, you get a jar of 100 differently colored balls, and randomly mixed, they make the next generation. You've got all of those differently colored balls represented in the population. When a population goes through a bottleneck, you put your hand into that jar, you pick up a handful of them, and that becomes the breeding population for your next generation. So you've lost all those other colors. And over time, this can happen multiple times. So you start losing genetic diversity every time. So do some species exhibit it more than others? Absolutely, right? Across the animal kingdom, some do. We work on, primarily we work on indoor urban pest insect population genomics. Species like bed bugs or German cockroaches, which are globally distributed in urban indoor environments, have some of the lowest genetic diversity we would ever expect to see in an organism. They're almost clonal whenever you find in populations, because like with bed bugs, they're founded by generally a single pregnant female that's hitchhiked a ride in your suitcase. She gives birth, those offspring interbreed and backcross with her. So if you're unlucky, you pick up one or two of those and bring them somewhere else, and they keep going. So we have colonies in my lab that were originally collected in 1973, and they've never had introduced genetic diversity to them. So there's 52 years of inbreeding, 6 generations a year, and yet they're thriving. Some species do well with that. So the reason they do well, and this relates back to some snake populations, is that we always think about inbreeding as being really bad. Inbreeding is not necessarily bad, because what can happen very quickly with inbreeding is that you select against or you lose those alleles at genes that are detrimental. So an allele is a variant of a gene, right? So if we think about, I'm trying to think about an example of it, right? If you're into boas, you've got the leopard, you've got the onyx, and you've got the Costa Rican black. They're all genetic variants of the same gene, right? They all produce a leopard-like animal, but they're likely to be locality-specific mutations. So think about those 3 different mutations, A, B, and C. They would be considered different alleles of the same gene. All right. So with inbreeding, if one of the alleles or 2 alleles at a given gene are detrimental, they're lost very quickly because those animals die. And whoever survives doesn't have that detrimental allele anymore. So therefore, through inbreeding, you can actually very quickly select for individuals that are more fit than you might predict by chance. Alternatively, however, if you're unlucky, you could also go the opposite direction, and you could fix for those deleterious alleles, eyeless, skinless, or scaleless, all those kind of things, right? So they can be detrimental. But some populations over time just naturally lose or purge these deleterious alleles. Great examples of that would be boa constrictor or boa imperator on islands, right? You've heard of coral key, lagoon key, maybe west snake key, other island boas that are found from really small populations, and yet they thrive. What's likely happened is over 16,000 years of isolation, those individuals that got to the island and bred, any of those with deleterious alleles died, and those that didn't have them survived, and they thrived in that island environment because it's relatively stable. But other ones don't do so well. Like I've heard that hognose don't do well with inbreeding, right? Well, so sorry, let me interrupt that 1 second. That doesn't mean that you can turn around and then inbreed your boa constrictors for 10 generations and expect because that's not a natural event where things are being selected against, right? What do we do in the hobby whenever we get that really pretty animal that's born but is barely fighting to survive? We do generally do everything we can do to get that thing to survive, because it's the nicest example, right? We assist feed it, we'll baby it along, and then we want to breed it. In the wild, it never would have survived, right? So those would have been lost from the population. So we don't, in captivity, subject our populations, our animals, to the same stresses and selection pressures that would happen in the wild. But with hognose, I've heard that even just with a handful of generations, you start seeing genetic anomalies. Is that right? Or some problems?
Yeah, I mean, I think it's the thing is, like, if you start doing some here I'll say this, with like captivity, we're talking about like artificial selection, and artificial selection can sometimes not be the best. Like with hognose, it's like pretty funny how like bug eyes and short faces within certain projects can pop up.
Mm-hmm.
And I know my genetic diversity is pretty good in contrast to some of these populations in the wild. There's some population. There's an isolated population in Missouri, an isolated population in Iowa, and they've been in Illinois. There's a couple, and they've been fragmented for like 60 years at least. Somewhere around there, 40, 50, 60 years. And some of these areas have a population of about like 200. Like the one in like Illinois, the one in Missouri is probably less. It's gotta be less. And they don't seem to be too affected by it. Now, in captivity, I have some fairly inbred hognose, but I'm not going like F2, F3, F4, like filial generation, like brother to sister, boom, 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 boom. I have like these little, like, I'll give you an example. Like my lemon ghost stuff, I've been working on from the time I was very young. And I still have a lot of lemon ghost stuff, and I still have like ones that are like actually pure lineage. And I'm getting to the point though where I can no longer actually really technically have any pure lineages. Like I haven't really made any pures in certain like this year. I don't think I made like I think I got some Woma pures lemon ghost. But anyway, that's beside the point. When I'm breeding like my lemon ghost, what I'll do is like I'm working on a project. I'm selectively line breeding them, and I'm working them to like other like really high yellow hognose initially, and that's how I'm founding this project with these high yellow hognose. And I find another unrelated high yellow hognose, and I have a few of those. And anyway, so I start breeding them, and I kind of like breeding like, you know, like a little group here, and I'm breeding them in a direction, and I'm breeding a little side group here. And then at one point I'll start crossing them. And then once I have these like kind of like cousins, like I don't really have any problems. I think like looking at these populations, as long as you're artificially selecting them for what I like to go for is good anatomical structure, because that's one you know, what we want. Also, it's, you could tell it's a healthy animal. It looks the way it's supposed to. Like, we want a wild Nassicus look. We don't want pug face. We don't want bug eyes. We don't want short bodies. And so anytime you start getting those, you just want to get rid of them. Because like you said, you know, it's the thing is, it's like there's these alleles that start becoming more common that are controlling these like poor traits. And if you have some like this, like there's one guy's like they made some outcross Loma Lemon-Ghost. And I would say, you know, them being outcrossed, it's like, okay, that's a good start. But then he started getting some cool looks, and he kept breeding them, and they got somewhat inbred. And they got inbred to the point where it was starting to be a little detrimental. And so now you have these ones, even when you outcross them, those characteristics you don't want kind of pop up as soon as you cross them back. So it seems like if you like, and that's the one way I've been able to selectively line breed hoggos and keep my stuff, like what I would consider is like technically they're selectively line bred. They are line bred. They are definitely, if you're going to be technical, they are inbred, but and that's what we're kind of talking about, like inbred, like you said. And I really like that you said that automatically does not mean a negative. It's like a lot of people hear brother to sister or half brother to half sister, and it's inbreeding, it's bad. They kind of, you know, anthropomorphize or extrapolate from, like, let's say mammals or like humans, and they like, okay, this is bad, so in reptiles it's bad. But you know, yeah, so as the hognose, it's like if I have like a few different like groups, ideally like base groups, I call them base groups. If I have like 4, 5, 6 like base groups, and I'm selectively line breeding something that's a polygenic line, like reds and yellows, I could get them pretty inbred, it seems like. But you know, they're partially related, and then they're having enough babies too, where I think there's like genetic diversity, because they you know, they have a decent amount of babies, and that's one. And let me know if you know anything about this. And I think this is an interesting one, because it's kind of like you're talking about the bed bugs, and then more advanced organisms, let's say like rodents or like largemouth bass. If you put 2 largemouth bass in a lake and they reproduce, but they have thousands of babies, so that's probably enough to make all this different genetic diversity. So when they get called, there's a small pool, but you know, you could have a largemouth bass population in a reservoir for eternity, starting with just 2. And rodents, I hear the same thing. Like, if people start with like a male or female rodent, like, you know, rat or mice, and they breed them, and then years down the road, they have this huge collection, and they're all doing well.
Yes, the rats and the mice one are interesting because, you know, what happens is that we work with rodents and we get a strain that works really well. They produce lots of offspring. They're healthy. They live long lives, and they're highly reproductive. And what people think that they should then do is introduce fresh blood into those to kind of freshen it up. But very often that can do the opposite and cause the rodent populations to crash, because with inbreeding, it can also fix beneficial traits. So as I mentioned, you've lost those deleterious alleles that could only be very mildly deleterious or very mildly problematic. But when you've lost them, you've lost them. And therefore you've fixed genes into good kind of gene complexes. If you add fresh bloodlines to it, you can reintroduce those deleterious alleles back into the population unknowingly. And so what you sometimes see are these really healthy rodent lines that have had new bloodlines introduced into it, and then the mice collapse. It seems to be very prominent with mice, not so much with rats, but very prominent with mice. I get multiple people calling me every year about their mouse line has collapsed because they thought it was a disease that was introduced, and it was likely just it was called outbreeding depression. So you can get inbreeding depression where bad things happen, right, due to inbreeding, but you can get outbreeding depression where bad things happen whenever you outbreed, because you're breaking beneficial gene complexes.
Interesting.
But indeed, the more offspring you produce, the more likely the representation of genetic diversity across the individuals have. So you think about a snake, A snake has got like a ball python or a boa. I think their genome size is about 2 gigabases. So what's that? 2,000 million bases, right? Yes, so about 500 million. So about 2,000 million base pairs, right? That's 2,000 million base pairs of variation potentially. So with recombination that occurs, and whenever eggs and sperm are fusing and so on, you get genetic recombination. There's lots of potential to make all of these different variants. So the more offspring you've got, the greater the distribution of the genetic variation across those from the parents. And therefore, if you cull those down to a small number, you're likely to have still a lot of individuals that got lots of genetic variation. Whereas if you've got 6 offspring, there's a much more limited amount of variation that's going to be distributed across those 6 offspring. So yeah, so the more offspring you produce, the more genetic diversity you'll preserve from the parents, and therefore the greater the likelihood they're going to survive bottleneck events down the line.
Yeah, it seems like it's got to be evolutionary too, because it makes sense. Like some of these snakes aren't moving around a lot in ranges, and they do pretty well with inbreeding. Like you talk about rodents, they're doing well with inbreeding, and it's just something that through evolution, it's actually they've adapted and evolved to these kind of breeding patterns. And to go just a touch real quick on the hognose, it kind of didn't answer it completely. I've only ever gone up to like F3 with hognose, and I did get some like short bodies and kinks. But the thing is, like, you could get those with like really unrelated outcross hognose too. I've never done it enough. I think if you took wild hognose that were really strong, I wouldn't be too surprised if you could go to like F5, F6, and or even do like, I mean, that's the thing too, though. That's like purposely doing the most severe type of inbreeding. But yeah, my hognose, like the ones where I have at least a large base, they do pretty well. But I did want to mention like that I've gone up to like F3, and I don't think nobody's really keeps track of that, and any more people are so afraid to do it. So there's very limited information on that one. But then like let's say ball pythons, like they have low genetic diversity. We only really see problems, Dave, with like just like certain mutations that have like known issues. Other than that, ball pythons, they seem to be like really, really good. They're like healthy, strong animals.
Yeah, they're almost doing better now than they did 10 or 15 years ago. You don't necessarily see all the fasting issues that they went through. They're easy to start from offspring. They breed in a wet paper bag without cooling. They're going to an aseasonal breeding, which it's part of that could be due to loss of a certain amount of genetic diversity. Part of it can be due to fixation of beneficial alleles. But without sequencing large kind of groups of individuals, we don't really know how that's coming into play. You were right in your last podcast. You talked about all of this genetic diversity coming from lots of different places, and that's definitely happening. The problem is it comes in from lots of places, but then it gets fixed and then goes through bottlenecks really quickly, because how often are new genes coming in, right? And then if you look on something like Morph Market or Fauna Classifieds or whatever, you can look at the number of people selling clown ball pythons or puzzle or whatever the fancy desert ghosts, whatever the new mutations that are all hot for people. Everybody's working with the same genes, and that means that they're all coming from low numbers of finding individuals, which means that genetic diversity is actually collapsed to a certain level with those already. So if you get a clown ball python from me, I don't breed ball pythons, but say I did, and you buy one from someone else down the road, it's very likely that they've shared a pretty close recent ancestor, right? And therefore they share a lot of genetic diversity already.
Yeah. The one thing, the ball python collection, though, there is a lot of large ones. You know what I mean? It's like how many clown ball pythons are being produced a year, or like other ones? Because then the other thing too, Warren, this is and you'll be able to touch on this and tell us about this. But it's like when I was talking about like largemouth bass, like they have like thousands of babies, and just through the sheer number, there's genetic diversity. You have like genetic recombination occurring on all these different animals. You're having that effect with friggin' clown ball pythons, because like how many clown ball pythons are being produced? Oh, yeah. So they are bottlenecking, because it makes sense. Like, okay, you have all these different like locales where like, let's say they got like a DG from here, clown came from this locale, orange dream. And as people try to start crossing them, there is some like unrelated scattering genetic diversity there from different locales, but they are starting to then linearly line breed, so they all start to kind of diverge, and you kind of bottleneck them, like you're saying.
And we can trace it back to the actual individuals that started it, and that's been in pretty recent.
Is that accurate? Is that accurate? You could trace it all the way back?
Well, we just know even, right? So we know when Tracy had the first clown ball python.
Sure.
Right? So we know a date whenever that lineage started. And that, at the beginning, that was all probably a clown bred to wild type. And we know kind of for the first couple of generations how that would have been bred. And there would have been outcross because it would have been a clown to a wild type. And then people would have bought all these heterozygous offspring, but they would have bought pairs.
Right.
So and bred instantly, right? So they would have gone through an increase in genetic diversity due to the outcrossing, and then start inbreeding. And that probably happened for multiple generations, that inbreeding, right? Whenever people bought head to head or clown and a head all from the same clutch and did that for a number of generations. But then there's a new trait they decide they want to breed into it, and it adds genetic diversity. But then to fix those traits, they then need to inbreed a little bit, and so on. So you go through these bursts of genetic diversity, and then rapid collapsing again of genetic diversity by buying siblings or close siblings and breeding them.
Makes sense.
So we could probably model that depending on traits, but there's certainly there appears, assuming these different genetic variants are coming from lots of different areas, that it's potentially increasing lots of genetic diversity each time a new trait comes in. But very quickly after that trait has come in, we're collapsing genetic diversity because we're inbreeding to fix those traits, if that makes sense. Right?
Yeah.
It's a bit different if it's a dominant trait where the heterozygous and the homozygous have the same phenotype. But if it's a heterozygous if it's a recessive trait or an incomplete dominant trait where there's a homozygous form that is different, then inbreeding has to happen for us to then fix those traits again. So you go through outbreeding and then inbreeding and then inbreeding and then outbreeding and then inbreeding and so on. So you can expand and collapse genetic diversity pretty rapidly.
Yeah. The comment you made before about how ball pythons were more trouble feeders back in the day, now they're a little less. You think that's just from keeping the strong feeders and just simply breeding them for years, or is that just something where they got more used to being in captivity?
I think it's through selection for animals that establish better. There were studies on foxes in Russia a number of years ago where they were able to take wild foxes, and within 6 generations they had tame foxes that were different colors, that had floppy ears, that were pleasant, they weren't aggressive, compared to the wild type. And that's just, it's what we're doing is we're domesticating animals. The whole idea about domesticating something is that over time it becomes a better feeder, it grows faster, it puts on weight faster, it's more reproductive, because we're always selecting for those traits, right? If we produce a litter of ball python or a clutch of ball pythons or a litter of boas, for example, and we want to hold animals back for the next generation, what we will do is we'll do 2 things. One is we'll select the nicest ones that we think are going to be the best, but ideally we want the best feeders, because they're the ones that are going to take off and get going for our next generation. So therefore we are artificially selecting for traits that are suited to captivity. And that's why, a lot of these ball pythons nowadays are just much better. They're totally different than what we had 20 years ago. I remember 25 years ago, 30 years ago, ball pythons were a flipping nightmare. Even captive farmed imports were tough to get going. And whereas now, you can take them right out of the bag and they'll probably eat within a week, probably on defrost as well.
So, on that point though, over the years, I don't know many ball python breeders that held back certain things necessarily because of feeding, because a lot of times, we're all chasing that new color. And if we hit it, whether it's a good feeder or not, a lot of guys will find a way to make it work. And I even remember stories back in the day, and I'm sure there's still some current ones, where people will assist feed, let's say, a male ball python that's just never been a good feeder to get it to breeding size. And I know people that successfully get those animals there. And I wouldn't necessarily say the next generation producer that one is the same feeding habits as the father was.
Yeah, right. But that's not necessarily going to be happening every time. So over time, unintentionally, we are selecting for traits that are suited to domestication.
No, that makes sense. Yeah, it's going to be a slow process, because I work with a species that's notorious or has a bad rep for being a difficult feeder for when they start out. And I think it's like, hognose is definitely a misconception. And, I was working with hognose from, I got my first one when I was 9 years old, very young. And my first one was a wild caught one. My first few were is hard to get captive borns. I don't think it was like until my fourth or fifth hognose I got a captive born one.
Mm-hmm.
I was mainly getting wild caught. And the one thing is like, yeah, they're definitely better feeders now than they were like about 20 years ago. Because by the time I was 15, 16 years old, I was like hatching like 50 or 60 a year.
Yeah. I
will say there is another aspect to it too, and I'm sure it's just ball python people too. It is a small aspect to it. Is like we get better at our presentation and our ability to understand the conditions that the animal might want to eat in or the presentation of the meal. So I've gotten a lot better at like getting hognose to eat with certain tactics and stuff, and whether it's the setup or just my overall presentation or the prey type, and then knowing like exactly what to use for scent and stuff. And over the generations, I've actually with hognose, usually I am keeping the best feeders because I don't want a hognose that sucks at feeding to keep back as a breeder. And some of it has to do too with their disposition, because a hognose that's more defensive can sometimes be harder to feed because it's put off by human presence, and your approach can set it off to not want to eat, and then you might have to leave a meal in there or feed it live, and so they can be a little trickier. And so I've always kept back like my best feeders. It's very rare I'm keeping back a poor feeder. And sometimes you see something that's interesting, Warren, and this is going to go back into like these are like just some genetic diversity. It's kind of actually what you're talking about. This might be a bit of an example. I've had hognose, like really, really good feeders, like very unrelated, breed them. And the babies are fucking horrible feeders, like the worst goddamn feeder. It actually just recently happened. It's really important. It was from a moonstone male. My moonstone, my lavender stuff eats really good. I don't have much problems with it. And I bred it to actually is a Swiss chocolate, and they're like kind of like they're okay, but the one I bred it to it was always a good feeder. And man, those babies were nightmares. They were my worst feeding baby hognose I've had probably since the leucistic stuff. So,
yeah, well, we're assuming that feeding behavior is related to one gene, and it's probably related to lots of different genes, and therefore the genes that are at play in one individual doesn't mean they'll be the same that are at play in another good feeder. So whenever you cross them, you break those gene complexes, essentially, then you can make poor feeders.
Yeah, I seen it. You had a whole clutch, man. It was like 14 babies, not one good feeder. But even my good feeders, like my red albino stuff, is some of probably my second most line bred project, because it started with red phase stuff that I got from Craig Trumbauer, and then incorporating extreme reds and purple line reds later on, and those feed really, really good. But it's still every now and then it's like, because the thing is like like I remember I actually read about that study with the foxes that they were selectively line breeding, and then they kept the ones that had the more pleasant disposition and weren't afraid of humans, and within just a few generations. And I remember hearing about that in the floppy ears. Is very interesting. The one thing is though is like the hognose and snakes and stuff. It's like I don't know exactly, but I can go take from like what I've experienced. And I see like fossils of hognose. Like they had fossils in Nebraska about 10 million years ago. Heteroanascus. And actually Nick Mutton told me, he goes, well, back then, he goes, it actually would have been, even though the fossil looks identical, he goes, it would have been a different species. Yeah. That's how I was like, oh, that actually makes sense. I didn't think about it like that. But they pretty much were unchanged for so long because it's just very efficient, and it works in their habitat. But anyway, what I'm getting at is like, over millions of years of evolution, sometimes it's like, even though things can change pretty rapidly, certain things seem like not completely change, right? Because like, I've been selectively line breeding my best feeder red albino hognose, but still 1 out of like 20, I'll get one that's like a, bitch of a tough feeder. And it's not that it doesn't want to eat. It wants a particular type of prey eat like lizard. It wants to eat like usually those ones you give it a small like if you every now and then you'll get these ones that don't want to eat absolutely anything. But if you give it a small lizard or frog, like if I give it a tiny leopard gecko, they'll like almost always eat it. And so it's not a bad feeder. It's just not identifying a mouse or even a scent, whatever I sent it with, or even a live mouse. It's not identifying it as a prey item. And that's from multiple generations of good feeders. But still, every now and then, it's like I get one or two. It's like, okay, these ones are a bit tricky. So it seems like, it is going in a direction. And I've been doing it for like probably selectively line breeding my best my red phase are like the best feeders. Been selectively line breeding those for about, I'd say, almost 10 generations, probably something like that.
Wow. Yeah. It's just behaviors are definitely heritable for many traits. It's just in what way they're heritable. It's unlikely that there's simple recessive traits for many things.
It's a culmination.
If they're polygenic, then as you line breed, you're still still not fixing all of those genes for in a homozygous form, right? So therefore, they're all going to be good feeders. But also remember that for every generation, for every gigabase of DNA, so for every 1,000 million base pairs, you get around 20 to 40 new mutations per generation. So as you inbreed, you are still adding small amounts of genetic variation. And where that genetic variation occurs, we don't know, because there's some recent work that suggests that mutations are not necessarily random, which is why we see the same mutations sometimes pop up in different localities and so on. So there's parts of the genome that might be prone to mutation. We're still that's the areas that are still being explored, pretty exciting areas of research. So polygenics and mutations are still things that are going to cause those odd anomalies to pop up here and there. Like I was very lucky. I bred my first hognose, I think it was in 1995. And from one pair, I got two clutches of 12, 23 hatched, one didn't, and it was because the baby was kinked. But all of those babies, they shed within a very short period of time and all fed on defrost pink mice, without any problem. It was kind of amazing to get those things, to see how fast they took off, because I was expecting, this is going to be a nightmare given their kind of frog lizard origins, feeding origins, but they were really easy, and that was 30 years ago. And therefore they couldn't have been that far from wild.
Oh no, not at all. You probably, they've been like one generation from wild if they if not captive hatched. There is one thing, the northern locales eat way better than the southern locales. Like your Lubbock County, I mean, it's not that they're all like that. You can get some stuff from like Southwest Texas and like even New Mexico, pretty good feeders. My one friend, he actually had some wild caught Mexican hoggos, which Mexican hoggos are notorious to be even tougher feeders than Nassicus. And he goes, yeah, everybody told me the Kennerli. He goes, man, they're a lot tougher. And he's like, I hatched out I think it was like 6, and he said all 6 ate frozen thawed pinks, no scenting or nothing right away. He goes, oh, I probably got lucky. He's like, well, those individuals, they're good. And so, but the northern locale hognose, it's possible you had those, or you had some really good ones. And
no idea back then. It was just picked up in the store and just as a Western hognose snake, and then got another one as a Western hognose snake. So we'd have no idea at that point, especially in Northern Ireland, where those things came from or what generation captive bred they were. It's thinking about kind of locality variation in feeding behavior. I've always thought about it and wondered whether it's in relation to prey density in those areas. So are we seeing more mammals in northern areas? If you got higher densities of mammals and therefore higher densities of mammals that are nesting and producing offspring than in the southern areas, then there might be a greater chance of those female or those offspring hognoses to encounter a nest of baby rodents.
Sure.
Start feeding versus in the I've been in over in Arizona in the Chiricahuas at Bob Ashley's place, and around his area you'll find hognose snakes quite frequently. But you see a lot of rodents around there actually, but you see a lot of lizards. So I'm curious whether they're primarily more lizard focused or whether they're more primarily rodent focused. There's definitely not a lot of amphibians whenever I'm out there, so it'd be interesting to see how they feed.
Is babies, they like, even if you go to like Illinois and Minnesota, like actually if you go to look for like Nassicus, pretty much like anywhere you go, you want to make sure there's lizards there too. And the babies, they find they eat a lot of little lizard eggs, whether they're skink eggs or like something like a 6-line race runner. And they'll eat small lizards too, hatchlings, and then the eggs. They do eat some rodents. Most of the studies though showed that it was like around like 6%. Like they eat like prairie voles and stuff, and it's actually more like the juvenile hognose because the babies are kind of probably gonna have a tougher time, finding a nest of like fresh pinkies. I'm sure it does occur. I think the reason the northern locales feed better is because they have a shorter window and period to eat, and they have to be a lot more opportunistic. So they're a lot more aggressive when it comes to and they're a lot more open to eating whatever they can find.
Yeah, that makes sense.
And then these southern locale ones, they actually because the thing is too, there's some I mean, there are some northern locale hognose with like high density populations. Like the Illinois population is pretty dense. That little Iowa population, I think, is like pretty dense. But there's lizards there, and there's a lot of it's along the Mississippi, and there's like a pond and bog area. So there's a lot of turtles. So there's a lot of female turtles laying eggs and a lot of food for the adult hognose. And I think and there's like race runners in both spots. And then in Minnesota, there's some pretty dense populations. But if you go to the dense populations, it's like really prime prairie habitat, and there's a lot of species diversity. And then you go to areas like Montana and stuff, and then the hognose density seems a lot smaller. And it makes sense because certain areas it's like okay, there's not as much food here.
Yeah. So to go back to, I got two things to bring up from that. One is you mentioned about these populations that have been contracted for about 60 years, but they seem to be still doing well. Well, 60 years is only about, 10 generations in a hognose?
Yeah, right. Yeah, it's not a ton.
They're probably breeding every 3 to 4 years. Maybe start breeding maybe at 3 years. Maybe males at 2, depending how well they've eaten, but females are probably 3 years old or 4 before they're
3, 4, 5. Yeah, I'd say so.
So you're looking at kind of at a maximum of 10 generations of population collapse and inbreeding going on. So, and given the size of it, there's probably a reduction in the amount of inbreeding relative to the time. So they're probably still pretty safe. I wonder, if you came back in another 100 years time or 200 years time, if those populations are still small, and they will be, they'll collapse further. What we're going to see. What I would love to do is to be able to collect samples from those and to undertake genome sequencing to actually look at how contracted those genomes are, because the cool thing is we can often go to museums and find specimens that have been in their museum collection for many decades, sometimes a century or more. Like we're sequencing the entire genomes of bed bugs that died 120 years ago. From there through to the present day, to be able to look at when mutations relating to insecticide resistance appeared. But it means that we can relate current populations to past populations, which is really cool. So with something like these populations that have collapsed over the last 60 years, you could look at the amount of genetic diversity that was present pre-population decline versus levels of genetic diversity present now, and relate that to the number of generations that they've gone through. So that would be kind of cool.
Yeah. And then the 60 years too was an estimate, because I should actually look a little, it'd be kind of tough. Like for certain ones, like the Illinois one, it's an estimate. The one in Iowa, you could probably actually figure out based on the company around it that, developed the area. You could see when that building was there. Sure. Now there's two spiders, like in Iowa, I know Zach Loftman, he's studying the hognose there. Mm-hmm. And then the Missouri, that population's like pretty small. The habitat, Dave's actually been there. He's gone up there to herp. I'd have to see it in person. Well, not really. I mean, I remember I've looked at it Google Earth so many times that I measured it, and I can't remember. I know it's not very big, and it seems like the Missouri population, because I was talking to a guy at Reptile Talks, and he was telling me how those are gloidai, and I'm like, yeah, I was like, it's not a subspecies. I was like, this is kind of like a locale type, and certain ones have that kind of the gloidai, or what they call the dusty hognose look.
Yeah.
He goes, I don't know, because I found one there with different head structure. I was like, their head structures always look typical to me. And he showed me one. I'm like, oh yeah, that is different. I'm like, man, I think that's from the inbreeding though. I was like, because that's
yeah, head structure is one of those things that goes. So again, if you go back to that, you go back to that Russian fox study, one of the things that they showed was that kind of the nose kind of squishing in and head structure changing, which is of course what we see in domestic dogs, right? Over not a huge number of generations realistically when we think about it. So
hognose too.
Those things to go. What was that?
Hognose too. It's so funny because like hognose, like you start like if you do like a little too much inbreeding or like where you're getting the negative effects, you get the bug eyes and you get the short face and you start getting these poor anatomical characteristics around the head. So it's interesting that this first because that's why I wanted to jump in and say something. I got excited there because you're telling me like it's like first thing they see that goes to the foxes, they start getting like floppy ears and the structure of the face changes. And with dogs too, it's like that happens in a hognose
and body size depressing.
Yeah, that's interesting.
Yeah.
So that hognose spot you're talking about, like the actual protected area, I want to say is like 200 acres, and it's just all sand dunes.
Mm-hmm.
But when we stopped there last time, we did pull up iNaturalist, and we did find pins on the map, like a mile or 2 miles away on like the road. I think it was roadkill. So where a big part of the population probably is in that area, they're still bleeding out into the neighboring areas.
Yeah, there's like one little area I think like next to it that they can exist. Some of those pins though, Dave, is probably because like at iNaturalist they kind of obscure them a bit, so they'll make them look like they're somewhere where they're actually not. Even though it's like you can only find those hognose in that one like little area. So, but it'd be interesting. That's why I was mentioning it. Those small populations again too is like how much would it cost, Warren, to do a sequencing of like a population and look at it and basically get a read on how related they are.
About for a population, probably about $5,000.
Okay.
Not including, that's doing it without assuming kind of salary and for technicians and stuff like that there. But cost for sequencing about $5,000. Yeah. Gotcha. So just through either whole genome or RADseq, probably about 4,000 to 5,000. So not a lot of money, not to be able to kind of determine levels of genetic diversity within populations. But it can also be done in captive lines, right? To be able to understand amount of genetic diversity within certain captive lines or captive, so if you've got your line A and your line B and your line C, you can look at the amount of genetic diversity within those lines and among those lines to start allowing you to select or to optimize your breedings, to make sure you're optimizing outbreeding and minimizing potentially detrimental inbreeding. Go ahead, Dave.
For what Junior's asking, and also, how many specimens would you actually need to find for a study? Because I will say, I know guys that go to the spot 3 to 5 times a year, and you kind of find one a year if you're lucky.
That's the biggest problem. For population studies that we're doing for many things, we're sequencing dozens to hundreds of individuals.
Oh shit.
Yeah, so that's the biggest problem with snake stuff, right? So, the work that we've done in collaboration with other researchers who work on Hog Island boas, they can go and they can find 30 or 40 animals pretty easily as they go as a research team and they spend a week really intensively sampling. But for things like hognose snakes, I keep meaning to email Zach to say that if he ever gets hognoses, to take a blood draw or a tissue clip and store it, and then eventually over time he might have 20 or 30 of them that we can run.
He should, from the sounds of it, where he's been at that I think they lay out boards, and he said that he has students and they have a bunch of them walk in circular patterns to find them. And I mean, knowing that those because the Iowa spot reminds me a lot of the Illinois spot, which is it's just on the other side of the river but further north.
Yeah.
There's actually seems like they're healthy populations, and so I think Zach probably's come across at least that many there. I don't know if they're taking scale clippings or what kind of samples they're doing.
I'll need to check because that comes with its own issues when you work with a university. So if you work with a vertebrate, you have to get IACUC protocols approved, which is basically they can authorize that you can take blood draws or tissue samples or euthanize specimens. And especially so that means if we're doing lab studies with rodents, for example, or with snakes, they can regulate the number of individuals that we're using. If it's field collected, especially where there's populations that are of conservation concern, that leads to lots of problems, and paperwork can take months or sometimes even years to get processed.
Yeah.
So but Zach will be in a bit of an easier situation because of the size of his university and his ties to that program. Whenever I was in Oklahoma, the university that I was at there, I was on the IACUC panel. So it's a lot easier to get protocols at least read and reviewed. Whenever you're not on the committee, you have to submit it, and it takes time to go through the whole chain of through the process, and it's a bit of a pain. and even some states, you need to have permits to be able to even pick up roadkill, right? So I was in Arizona just a few weeks ago at the Biology Pit Vipers meeting, and you're driving up the road and there's a smashed diamondback rattlesnake on the road, but you can't go out and pick, or a western diamondback, but you can't go out and pick it up and sample it unless you have permits to allow you to do that. If you even go over and touch it and Fish and Wildlife see you, then you can get cited. So there's all of these hidden complexities to make these studies very difficult.
Yeah. Yeah, so we need more laws and regulations against everything. Look at that. You can't even touch a dead rattlesnake.
Yeah, I know. So that's why the ball python stuff would actually be a lot easier, to be able to go down to Outback and say, hey, you brought in 300 or 400 or 500 today. Can I get, and in that bag of shed skins for 20 of them, can I have those shed skins? And they'll be like, yeah, take it. Because they're likely to, especially those random bags, they're just likely to have been whatever was there, just thrown into a bag and shipped. So they're giving you some level of diversity from across those populations. Yeah. So they would be really interesting. And if they could turn around and say, we got these from this farm, and we got these from this farm, and they're all in the same country, and we got ones from this farm, this farm, and this farm from different countries, it would allow you to start looking at genetic diversity across that range.
That'd be pretty interesting to see. I'd like to see that.
Yeah, if we were able to get the samples, I would fund that study. That would be a neat one to look at.
So at the end of the last episode, Warren, I'm sure you tried to hear me mutter through this question or idea, but with Hog Island boa's population almost completely getting depleted, I want to say there was a couple studies done soon after that where they found no specimens, and now they're actually starting to find them in the wild again. You saw the Dave Kaufman video. I think he discovered like 3 or 5 of them, but I think it was near like a water source where there's always, you're always going to find them. I heard there's a well and a waterfall. But what I'm bringing up is, so long-term effects of that species that's already an island species, I'm not sure how genetically diverse they are in the first place. As it starts to repopulate with the little population that was left behind, what do you think we might see as genetic anomalies? Do you think it'll make the species smaller? Do you think it'll change the overall, not the overall look, but do you think it could change the look a little bit depending on the ones that survived?
So my feeling is that the population never really got that bad, never really got that low. The reason for that is that with collaborators of mine, we've done work with Hog Island boas, and we're actually just about to start, hopefully, a new study of genomics of which are going to involve wild-caught Hog Island boas. When you go there, you'll be able to collect about 4 an hour if you go and you walk through any of the areas. I think the reason that the first couple of studies or articles that were published that said they couldn't find any boas is they looked in the wrong place. Where they're finding Hog Island boas now they're all in the trees. You don't find them on the ground. They're all small and they're all in the trees. And I think the first party studies, they were just walking along and looking on the ground, expecting to see a boa. But you go for the last 10, 15 years, the densities have been high. And we've got a whole bank of tissues that we could work through with hundreds of animals that have been sampled on the island. So they're not uncommon. I heard that the Dave Kaufman one, that's where he went with Quetzal Dwyer, right? I think they found 6 or 7 just by walking up, one little path, and they were all in the trees. But what you would predict, so say that population did go through a collapse and it got repopulated. So Hog Island boas came in to the country about 30 years ago is when we saw the high densities coming in. And then they disappeared, and the reason they stopped was because the Chaos Coquinus Islands became a reserve, and therefore they banned any export. There was at one point in time a number of animals that were smuggled off the island, and they were distributed to zoos. I think it was around 50 animals or 40 animals, and there was meant to be a second bag of them that were ditched, so they disappeared. They obviously died somewhere on the island or in the water, but there was about 50 animals came in. But given that the island was never necessarily depopulated, it's not really an issue. If it had been depopulated, whatever was left there is going to repopulate the island because there's still abundant food sources and so on. And what that's going to be made up of is the genetic diversity that was there in the prior to the whole process. So, for example, the animals that were originally collected, they tried to collect those that were not very speckled. They wanted the really clean animals and left the dirty, kind of muddy ones there. You're expecting the population to be founded from those dirty, muddy animals that were left. And therefore, you would expect that phenotype and therefore that genotype to be carried through the present day. But we don't see that. There's quite a diversity of phenotypic looks of those animals across the island. Whereas you go to somewhere like Lagoon Cay or West Snake Cay or Crawl Cay, and those animals are all phenotypically almost identical. There's little to no variation because of thousands of years of selection for a certain trait where they're ideally suited. Some of those islands are primarily rock, with some trees on it, so therefore they're dark animals, and they're primarily arboreal. If there's anything that's kind of more lush, you're going to see a greenish color. So, for example, Corn Island boas, they've got a much more greenish hue to them due to the island structure. But you'll see selection if any population goes through a bottleneck and is then found, a new population is founded from it. You'll see those populations later are going to exhibit more of the traits of the animals of the founding populations. But we don't see a lot of interesting traits in Hog Islands, right? We know there's a purity positive, right? So there's the purity positive Hog Island, and then we see the patternless back or the striped ones, and that's about it, really. Yeah.
So, inbreeding in general, I think back. I was told like some of the original bearded dragons that came in the country were very large animals. Over years and years of breeding in the United States, they've been getting a lot smaller. And also, we were looking through an old Honduran book the other day. And man, I think this Honduran in that book was like 7 foot, 7'6" or something crazy like that. I have never seen a Honduran in captivity that size. Is that another just a lack of like diversity? We've line bred them for so long that they're just getting smaller.
It's where the Hondurans were picked up from. So mainland Hondurans are big animals. But you got to also remember that Honduras, in relation to boas, includes all of the Bay Islands. So Utila, Cayos, Coquinus, Major and Minor, and a whole other bank of islands. And they all have specific phenotypes and looks to them. So the fire bellies, for example, came off one of the islands, they believe. So it's where those initial animals came in from. And it's also, you've got to be able to trust the animals in captivity that are labeled as Hondurans. A lot of animals that have come in that are labeled as Hondurans are very likely to be Nicaraguan farmed animals that were then, we'll call this something new and we'll get a little bit of extra money from it. So I've certainly seen big Honduran boas. The ones that we see that are carrying the specific traits that people are interested in are very small. And that suggests that they were likely an island population that was sampled. Or a mainland, but from one of the smaller Central American mainland populations. Yeah. So I've seen a Costa Rican boa in the wild. Whenever on one of my trips to Costa Rica, it wasn't far from the research station, and it was easily 6.5 feet, whereas my Costa Rican boas here are pretty much 4.5, 5, maybe 5.5 feet. It was a bigger animal, but also remember that the snakes are indeterminate growers, which means that they continue to grow throughout their life. They'll put on size whenever the season is good, but they also grow really well in the off season when they're utilizing that stored energy sources that they built up from before. So I've had a Sonoran boa that was easily 7 foot, but it was a really old snake. It was probably in his 40s. Yeah.
So I love the answer. I was talking milk snakes.
Milk snakes?
Oh, Honduran milk snakes.
Honduran milk?
It was a Honduran milk
Norm
snake.
has some big ones. Last time I was over at Norm's, he had a male Honduran that was like 6. It was over 6 foot. It was like probably, it was definitely pushing over 6 foot.
Yeah, but also you're going to see natural variation in a species. While most humans will be in the 5'7", 5'8", 5'9", 5'10" range, maybe a bit bigger, you also get those ones that get up to 7 foot, and you get those ones that are 4 feet, 4 feet to 3.5 feet. There's a natural variation or decline in size. So it's very likely that those kind of animals, one, could have been very old, and two, could have been at that upper end of the extreme. Frank Martin, for example, has a line of boas that produce giant boas. And he said that within his litters, he can pick out the ones that are giant and the ones that are not, just like an albino versus a wild type. So there's for hunter and milk snakes, for any species, you're going to find really big ones and really small ones, but they're going to be at the extremes of that tail. You know, you're going to see most of them sit within the bell curve, you know, of the normal size range.
What are your thoughts on the giant gene with Frank? Like, have you ever seen anything in person? Like, what are your thoughts on it?
I've seen pictures of the adults, and they're scarily big. I've seen animals that are from that line that are scarily big, and I've seen offspring from that line that are scarily big, where they're starting on adult mice as their first meal. So my feeling is that it's definitely a genetic trait. Whether it's polygenic or whether it's recessive, it almost looks like it could be a recessive trait, because of the way that you can breed it to a smaller animal and pick out individuals that are large versus small in the litter. It doesn't seem to make sense that it'd be a single trait, but you know, I haven't looked into it with enough focus.
That's fine. So Junior, you know, this is one that you probably remember because we've talked about it many times. But Moose, you remember Moose?
Yeah.
The
Oh
leopard
yeah,
gecko?
the giant leopard geckos. Yeah, that was one of I guess a debate, and I went on, like, I've talked about it on certain podcasts. Like, I haven't done a leopard gecko podcast in a while. But
Good.
yeah, the whole thing was when they first came out was I guess it was recessive, and then later he says incomplete dominant. I never bought any of that. I was like, there's no way.
No.
You know, there's so many you know, pairs of alleles that contribute to the size of an organism, and it's like, I don't know, it's like I'm never seeing like a gene, like a single allele, you know, mutant gene that is like, let's say, like in dogs or, you know, any reptile I've ever seen that, you know,
That's the size.
it does something to the animal's size. But I mean, once you see what they do with the giant leopard geckos, it's like, you know, they're selectively line breeding them. Like, they're like, oh, look at Moose, he's the biggest one. You want to buy one from the biggest one, you know. And then the funny thing is, when Moose came out, you know, he was like, I think 155 grams. Now that these guys have been selectively line breeding these leopard geckos, these big leopard geckos, very well. One thing is you need two things. You need to selectively line breed the biggest ones, and then also you need to feed the bejesus out of them, because they don't get big unless you fucking cram them with food, and they don't get heavy unless they're minorly at least obese, if not full-blown obese. But there was somebody at Pomona, man, they had like 170 and 180 gram ones, which are way bigger than Moose. And so now there's leopard geckos, and I think Steve Sykes, I think he might have the world record. He had one that has like 200 grams.
Yeah, because he had one, and the offspring of it was heavier than the parent, right?
As a baby, yeah.
But the other thing about it was, look at how long they're living.
Yeah, the Moose died at an early age. I mean, like, some of these ones, like I don't think many people are coming forward if they're dropping dead early, but yeah, any, you know, animal that you are feeding really heavy and then consistently like slamming with food with the purpose to get it as big as possible, you're shortening its life expectancy.
But it can also be related to pituitary gland growth hormone issues. So
okay.
What was that wrestler called? Remember, he was in The Princess Bride.
Man, Andre the Giant?
Yeah, so his was a genetic trait. That was a genetic anomaly, and they knew exactly how to fix that whenever he was young. Medication could have stopped that, but they decided he didn't want to take the medication. You know, so
worth it, buddy. Totally worth it.
There are pituitary gland issues that could be related to a single trait, a single gene, that can cause the overproduction of juvenile growth hormone that can then lead to gigantism.
Yeah, but usually, like Andre the Giant, it's pretty apparent. Like, it's affecting his anatomical characteristics. He's also like abnormally large. With the leopard geckos in particular, and for this, there's a cline to it. For one, they have to feed them really heavy, and there's also this cline. Like, you could breed giants and make ones that aren't even that big. Like, when I bred my giants, and with the way I feed my leopard geckos, my giants, none of them really ever got that big. You know, they're always just kind of like moderate size, because I was not really slamming them. But
is it the thought that they also came from a hybrid? Were they a hybrid or an integrate? Where they were a...
No, they were just. I mean, Macularius were initially kind of like, you know, I mean, they were all like mostly like, as far as I know, like the majority East Pakistan. Yeah. They definitely weren't really in any mix of Afghanistan, because the Afghanicus are small, so that wouldn't. But you know, like you have like the Angramanu, but those didn't come out till later, and you know they weren't brought in until long after giant lines were established. And based on everything I've seen and known, they're not high. They're definitely not hybrids, but they are mixed locale mutt macularis locale animals.
Yeah.
And there are certain locales that probably do get a little larger. You know, like the popular locales were like Fasciolatus and Montanus. And Montanus did kind of have a longer bodies, but you know, they're kind of like lighter individual animals. But you know, I remember we had this one female leopard gecko, not from giant bloodlines. She got to about 130 grams. She laid gigantic eggs, and all of her babies were big. Huh. And so, you know, we kept those back, and it's like that's how the giants like started. Like, oh, these ones are big; they make big eggs. And the thing is, it's kind of like, you know, I'd say it sounds like with the leopard geckos, it's not because it's pretty hard to control. It took them a long time to really selectively line breed them to get them to consistently get these pretty big ones, and they've done it a lot better now. Because I see like that one person at Pomona, they had, you know, multiple all in a row, 2 rows, very impressive giants. So it's like you're definitely seeing the results of a consistency there, regardless of, you know, how heavy you're feeding them. You could tell these were, you know, even heavy feeding aside, were still impressive or going to be very impressive individuals as far as their size goes. Yeah. But yeah, with when we bred like giant leopard geckos and stuff, is like it's pretty inconsistent, but it was still early on. So, you know, I feel like it's almost like allele patterns, not as like, because I think they're more controllable, but like in humans, you know, you could have 2 shorter people make a taller person. But if you start selecting all the people, that's why, like, you know, there's actually, you know, indigenous like people in certain areas, they're much taller, and in other areas they're much shorter.
Mm-hmm. Yeah, leopard geckos are interesting because, you know, what Ron Tremper put his through decades of inbreeding. You know, what was it, like the 42 generations or 47 generations or something absurd like that?
Yeah, he went like pretty far. I know he was talking about like, I don't know. I know he went to at least like with filial generations. Like I know he went to like a F11 or like maybe he went out like higher, closer to like F20. Yeah. And he definitely did a lot of like inbreeding. And I seen like a HQ Reptiles, they went up to like F10 and leopard geckos. And the funny thing is, as long as you were selectively line breeding ones that didn't have like kink tail, they were doing fine.
Yeah, like I think he said it was only in about the 6th generation that he started finding the ones that were kind of anomalies with stripes, or they were slightly different colors or whatever, right? I think it was after about 6 generations of inbreeding.
I don't remember that. I don't remember that. I definitely know he you know, I think he started with his leopard geckos. I want to say it was like the 80s, and there's a lot of imports. And
he got his through a zoo, I believe, right?
Okay.
Yeah.
Yeah.
Through a zoo that he wanted to then breed, but he probably brought in imports after that. Yeah.
There was the funny thing is too is like he, you know, popped up with the Tremper albino. The bells, they had the bell, you know, it's a you know, different mutation or non-allelic, and the bells actually got their, from what I understand, remember, their collection of leopard geckos from Ron Tremper, but it's like kind of more wild caught stuff, or like, you know, not far from generations. And then Jay Brewer, he produced the first blizzard leopard gecko. Yeah. And then he had the patternless, and so he had all these mutations like actually pretty quick. Mm-hmm. But then, you know, they're not bringing any more wild leopard geckos. I mean, they kind of are because they're select locales, but not like, you know, mass numbers of like, you know, wild type, like just like macularis. And yeah, so you had all these mutations pretty fast, and then after that, like boom, no mutations for a long time. You know, you had the Max Snow, and then you had, you know, a couple, you know, terrible ones like Enigma and Lemon Frost.
Yeah, yeah. But it's interesting that so with leopard geckos, most of the traits are polygenic. Is that right?
Yeah, there's a lot of polygenic. Like they're just like really naturally polymorphic. And the one thing that seems like in, you know, this would probably make sense. I think you agree here. It's like, you know, certain like reptiles, they have to have a lot more alleles, you know, for pattern and color, because that's part of their way to adapt and live in their environments. You know, it's their camouflage and whatnot, or, you know, predator deterrent.
Yeah.
Yeah, something like leopard geckos, they're heavily patterned, and they also have other like pigments. Seems like if you have something that's heavily patterned, or at least decently, that has like a level of like pigment coming through, there's more alleles that can be manipulated And things can be like made like of it, whether it's selective line breeding or you got something like ball pythons where they seem to throw a lot of mutations for some reason.
Yeah, I'd always thought we would see more mutations in leopard geckos that are recessive, but then again, look at corn snakes. I think most of the mutations in corn snakes are recessive, and then you only get a handful that are incomplete dominant or dominant. Whereas in ball pythons and in boas, you get a lot of mutations that are incomplete dominant or dominant, and fewer that are recessive. You get this weird switchover in the ratio of dominance, incomplete dominant and recessive.
I feel like colubrids as a whole is like that. It's a lot of recessive projects in colubrids.
Yeah.
Hognose too. There's the funny thing is you have the new gene Antarctic that popped up in hognose, and it's allelic with Arctic, and so it popped up in China. So those were it's like, again, like you're talking about mutations like that appear again, but they're like unrelated. Let's say like Mojave and Lesser, they're allelic, so they're mutations of the same allele, but they're definitely different. You could tell that, hey, these are not like they're not the same gene. They're mutants of the same allele, but they're different types of various or little variation of that mutant. And so we have the Antarctic hognose, and it's basically the same thing. It's it's the same allele. It's just a different type of mutation.
So it's just a different allele, a different variant of the same gene. That's kind of cool. Yeah, because think about the sort of Mojave and Lesser and all of those ones. They're ultimately breaking a product that a gene is making a product, generally a protein, right? So whenever there's a genetic mutation, it's stopping the production of that protein, or it's varying that production of that protein. So if you've got a Mojave and you breed it to a normal, some of them got that broken variant, and some of them, and the broken variant doesn't mean it's broken and it's not functioning. It means it's just not functioning to the same extent as a wild type. So it can either stop the production and produce the variant or the mutant, or else it can modify it and produce the variant. But whenever you've got a Mojave to a Lesser, you've got two variants. They're two non-wild type that are crossing, and they're sort of essentially two broken variants of the same, of two broken products in the same gene. So the Antarctic and the Arctic is interesting. I was looking that one up a couple of days ago. You can see that it's along the same lines as the Arctic, but it's just got variation to it.
Yeah, it does. And then the hetero Antarctic, hetero Arctic, it makes kind of like a super Arctic looking animal, and they vary. Sometimes they almost kind of look like a really weird distorted super Arctic, and then sometimes they look like a super Arctic that's like ultra black with like a white head and like shattering on the body. They're pretty unique. And then there's another thing too. The funny thing is too, I was like, this is just kind of like something fun to talk about. It's something that I've learned. It's like, when you see allelic traits, generally you kind of assume that, hey, since they're mutants of the same, gene, the same DNA code, that they have to kind of look similar. But then you get something like, like Enchi and Cinnamon, they're allelic and they look nothing alike.
Yeah, absolutely. Yeah, so the gene is the product is being modified. It's not, where that modification occurs. So it's making a protein, and a protein folds up into a three-dimensional structure, and it interacts with something else. But if that structure is not, it depends on how that structure is modified, whether it just stops it completely or else it makes a form that's slightly different. It has a knock-on effect, because remember that these proteins are actually having a knock-on effect to other genes and to other products and so on. So it depends on where in that chain that event occurs, can lead to a totally different phenotype.
Gotcha. Yeah. So when you see something like a Lesser and a Mojave, they have they're inhibiting proteins in a very similar fashion. Then you got something like Enchi and Cinnamon, and they're changing the protein structures very differently. Now it's the same code, but it's a very different kind of structure.
Yeah.
So when you're talking about the Bell allele stuff, wasn't there a recent thing that came out, and I didn't acknowledge it, where Butter and Lesser were proven to be different genes, not the same?
Possibly. I think.
Well, let's just say it's a yes. We don't know. We're not going to talk about it.
It kind of makes sense, because the original Butter, the original Lesser came into Ralph, and then a Butter, did that go into the Bells?
Yes, yeah.
And then a second Butter came into Ralph. from wild. So if you go back to his old, remember he used to, on his website, he had that diary that he published all the time.
Those are fun.
I think he mentions in that he got in a wild caught butter as well. And there's two different forms of butter, I think is what they're saying. Right? There's lesser is one, and then there's two different forms of butter, because one butter is like, remember he calls it, was it the lesser daddy or whatever? It produced a different variant.
Oh.
I think a butter, there's a butter as well. And then the other that. What was that, Dave?
Oh, he's breaking up.
Yeah, this guy and his high-tech internet. It looks like he's in the back of that car.
We might have to wait, or you might have to exit and come back in. He's talking about because Ralph originally got the original lesser that came in he called it a Platy Daddy.
Yeah.
The Platinum.
Yeah.
And then when he produced lessers, Dave was telling me about this. We were just talking about this, and he refreshed our memories on this one. And then when he bred the Platy Daddy, his first offspring, the lessers, didn't they didn't really look quite as extreme. So he called them lesser platinums because they were
to one of the siblings, the wild type siblings in the clutch. Then he got Platy Daddies again.
Yeah, so it seemed like Platy Daddy was like Dave was saying, I didn't know this was like a recessive mutation that was along with it that reacts with the lesser.
Yeah. Sorry, Dave, we lost you for a minute there, Dave.
Yeah.
No. All right. Just when we thought he was coming back. Yeah. You should jump back in. So we'll see. Give him a couple minutes. Yeah. So like I remember hearing about. Here's the thing. I'm not like, I love ball pythons. I think I like genetics. I think they're cool snakes. I'm not really a ball python guy. I have kept them and bred them, but I don't keep up too much. Like, I mean, I try to here and there, but there's just so much going on, as you know, and as everybody else, that's going to be listening is going to know. It's like, yeah, there's a lot of stuff going on. And I remember there's always arguments over the butter and the lesser because they were very similar looking, and people would say, well, the butters kind of have like a little more colorful look. But, that could always be too, in part, like, selectively line breed. Like, people are passively breeding for the nicer ones. And so I remember people would talk about like, oh, this guy has the best like pastel lines back when pastels were like more money. And when I was like in my mid-teens, and it was like, well, it's like the people that had the nicer pastels, it wasn't they had like a gene that was more pastel-y. They were just breeding them to prettier animals. So if you took away the pastel, the base animal was a lot nicer.
Yeah, what you got to remember with every gene that we're playing with every animal you look at, you're seeing the effect of that mutant gene and polygenics of lots and lots and lots of sometimes thousands of other genes that are playing a role in how an animal looks, whether they're slightly lighter or darker, more pattern or less pattern, the pattern stretched out or it's more compressed. It's not necessarily, so when in the case of those brighter pastels that you're talking about, they just happen to be those that happen to be the brighter ones, from other genes that were causing that. Yeah, so it is selective breeding and it's polygenics in play with genes that we're not necessarily trying to select for.
Yeah, it's pretty neat to see that because like with ball pythons, starting a polygenic project would be quite the undertaking because they're not the quickest to breed sometimes. I mean, I know people can get males to breed fairly quick, but sometimes females, like I know people breed females at like 2 years old, but sometimes they take longer. So a polygenic project with ball python is going to take, probably a good decade of like hard work to get some good results.
Right. You go buy a pastel and buy one or two female wild types, just fresh imports, raise them up and breed them. And you can guarantee that the pastels in each of those clutches are going to there's going to be a climb. There's going to be some that are really nice and some that are kind of crappy. Take those and line breed the best of the best and the crappiest of the crappiest. And you'll see very quickly within a generation or two that you're selecting for those variants that are brighter or darker.
Yeah. And so that's like
very quickly, but yeah, it takes time. Whenever, what's the generation time on a leopard gecko from hatching to reproduction?
You can get like sometimes 10 months,
Wow.
Usually like, I mean, I'd say like, some people like, they're not going to like hearing that. I don't know how many leopard gecko people are going to be listening, but like you can do like I've had male leopard geckos breed at like 6 months, 7 months old. When my dad and I were breeding them and we were, excited about doing projects, we'd, sometimes these males would get up to like 50 grams in like 6 months.
Amazing.
But our females sometimes like 10 months, but usually like you can just say safely a year. But that's one thing I learned a lot about selective line breeding and polygenics with leopard geckos, because you get such a quick turnover. And then since they are kind of naturally very polymorphic, like they throw a lot of variants, you know, you can see quite a bit. Now, every species is, you know, of course, different. So, but you can extrapolate a lot from, you know, selective line breeding leopard geckos. And like the ball python people are talking like, so we're talking about like breeding their prettier pastels. They can breed these mutations, like make like DG, like Orange Dream stuff, and these other ones, and passively selectively line breed as they're going to make these mutations. Keep picking out the prettiest ones. And I remember some guy, he posted a picture, and he did some combination, and he made like a multiple, like I think it was a triple het, and there was like I think pastel and Orange Dream in the pairing too, but he hit on a normal. You know, so he made the multi-het, but it didn't have Orange Dream. It didn't have pastel. And the guy was blown away because he was like, I mean, this is like the prettiest ball python. But it's a result of those prettiest Orange Dreams that people are making and all these other really pretty ball pythons. And I'm sure sometimes the hets, you know, sometimes you get some interesting looking hets, but it's just really bright, clean, really orangey gold, really nice white trim, normal ball python. And that's kind of like, oh, okay, they're like a lot of these guys were probably unknowingly selectively line breeding really pretty stuff. So I was like, hey, if you're actually making your ball pythons prettier, like on the like behind the scenes, as you're kind of picking out the prettiest of the prettiest for these projects.
That's exactly it. They don't realize that those normals tell you a lot about your line breeding that you're doing.
Yeah, and the polygenics is like, it gets overlooked a lot because it's like, okay, it's not like as impactful as a mutant, especially right away. But you can do a lot of crazy things. They look like koi carp. I think they did that in like what, 200 years, 300 years maybe, and it's like
or dark,
it's from wild carp, you know, that were like goldish and black, and now they have them like orange, black, and white, and like, you know, all kinds of crazy colors.
So what's the generation time on Western hognose? Is it females at 2?
Yeah, about 2 years is like the quickest. I mean, some people like I do some females at 3, but I can do a lot around 2.5. But males, I mean, I just had like well, this is a project that involves selective line. It's a lemon ghost sable, so and it has woma. So woma is a pattern that popped up out of lemon ghost. Lemon ghost is, you know, bright yellow, greeny, like, you know, selectively line bred hognose lineage. And I had this male that's a 75% from my basically 75% lemon ghost woma sable, and he's a really pretty one, and he's about 11 months old, and he looked like he could possibly breed, so I bred him to a female, and he actually locked. So sometimes you can get like a half a generation, you know, up quicker with the male.
We see the same thing in boas. You might get a male that's 18 months old to breed. You know, they're definitely going to go at 2.5, but you might get that 15 to 18 month old male to breed, and you're kind of you're sidestepping a little bit and, you know, halving a generation time.
Right. Yeah. And the boas, there's I know there's like the Red Rum Project, and there's a few other pink ones. I know they I think, oh man, what was I think they used to just call them salmons back in the day. See, I don't know.
The hypos, the salmon hypos that's salmon hypos and orange tail hypos are what people now just, whenever they refer to hypos, they just call them hypos. But they were originally 2 specific lines that came from the same founding animals. That the original hypo animals came in through David Hardy, and David brought them in, and he kept them in his office and bred them. These were collected in Panama, and he got a bright one and he got a regular one. He brought them home with him. He bred them and he just gave them away as brightly colored boas, and the other ones as normal boas, but he bred them for Panama to Panama. Eventually, some of those ended up with Rich Eiley, and Rich owned a pet store at the time, and a woman brought them in. She said, here's the parents and here's the offspring, and there's these really colorful ones. Rich tried to buy the parents, but she wouldn't sell them, but he bought some of the babies. And he didn't keep any back, he sold them, but a year later she brought them back in again. More offspring, and he bought those, and I think he bought the adults at that point in time, and he renamed them salmon boas. He did a genetic study with my friend Gordon Shewitt, and they kind of went through dozens of breedings to be able to show there was an incomplete dominant trait. but you get a cline, right? You get crappy looking hypos, salmon hypos, and you get really nice looking salmon hypos due to this kind of line breeding effect. The other line, which was the orange tail hypos, were just those that were Panama to Panama to Panama to Panama, generation after generation. So they fixed more of the Central American traits, whereas the salmon hypos fixed more of the Colombian boa traits. Yet they're the same mutation; they all derive from the same animal. But regarding the red rum, you'll find they're polygenic traits, and it's just like the East Bay Bavarian line, the RC pastel. You get a bunch of them that are producing more pastel colors or more red in the background, like the Russo reds and stuff like that there, and that's polygenic through selective breeding.
Yeah, that's nice. It's
probably similar to the high red hognose that you guys work with as well. Like they're polygenic traits, right?
Yeah. And hognose in the wild, I mean, you'll get some that are kind of already like decently red. They're kind of like a burgundy, rusty red. I've actually seen some, people got some pretty red hognose, and I mean that's kind of why you're able to get such red hognose within like originally it's probably like only like 5 generations, maybe even less, where they're pretty red. And even now, I mean that's one thing that's exciting about selective line breeding and within the reptile community. It's relatively young. I mean, how long they've been selectively line breeding, like these pink boas, like really well, like maybe 25, 30 years.
Captive breeding in general has not been going on that long when you think about it.
Yeah.
Selective breeding has actually been even shorter than that there in most cases.
Yeah, it seems like people are getting more serious about it now in the last like decade, 15 years. And so like even with hognose, it's like, okay, we're selectively line breeding them for pretty ones, like, or people were like in the like mid-late 90s. But they weren't as serious back then as like people are now. They're people now, like, there's a lot more people in the community doing it. People trying to raise them up a little faster. They're going a little more, I feel like, extreme, where they're like, getting nice ones and they're immediately like, all right, let's breed these to another one that like looks similar, that's a little outcross, or breed it back. Try to add some diversity, but just continue with this. So yeah, I mean, look what they've done with like koi carp, and what they've done with the domestication of things like rodents and dogs and cats in thousands of years and even hundreds of years. And the reptile community is relatively young. Like most of this is like a lot of people weren't doing much selective line breeding up until about like 20-25 years ago. That's like really started in the late 90s, early 2000s.
Look at leopard geckos. My first pet reptile was a leopard gecko, and that was 31 years ago, something like that there. And at that point, you could find leopard geckos, or if you were lucky, a high yellow leopard gecko.
Yeah.
Right? This is in Northern Ireland. I brought in the first Tremper line albino leopard geckos from Ray Hine in the UK, and I got them into Northern Ireland. And then just after that, I brought in what was called then a leucistic, but it's the Murphy patternless.
Right. Yeah.
They were so it was high yellow, wild type, Tremper albino, and Murphy patternless were really the only variants. And within a short period of time, a handful of years later, Ray Hines started putting out the tangerine or orange tail or whatever he was calling them. And you really started then to see the explosion in all of these color and pattern morphs. But for a species that is so globally distributed as a pet, 30 years ago, there was not a lot of variation in what you had as a leopard gecko. It was a leopard gecko, was a leopard gecko. And if you were lucky, it had slightly brighter. So it's amazing that when you think about hobby, it's so short. It's such a short-lived hobby so far, and yet the amount of variation.
Can you hear me?
Yes. Yeah, you're interrupting.
I can't hear you.
Really? All
right.
Oh, we lost it. Yeah, but it's amazing, and it's such a short period of time, the amount of variation that we've now seen.
Yeah, it is. And I remember Ray Hines came to Daytona a few times. I was like 15, 16, 17 years old. And he was kind of like, for a bit there, he had like a very impressive like sun glow. And actually that's where like, that was the rumor at, Urban Gecko. And he is known to have nice like sun glows in the early 2000s that, he got those from Ray Hines. And anybody that had a nice super hypo tangerine is like, okay, or sun glow came from Ray Hines line. And he was like, I guess he was the first one with a sun glow leopard gecko or Tremper albino, tangerine, the super hypo tangerine that he coined, like, okay, this is a sun glow.
Yeah. Yeah, they were his website was one that I would constantly lurk on 30 years ago, 25 years ago to see what he was producing. You know, because when you live in Ireland, you didn't have the opportunity to get all of the stuff that you had here in the US. You know,
Right.
much smaller gene pools to play with. Of course, that's changed now. You know, the UK and Europe and so on, they get as much stuff as we get here. But it was different. But I just think back to it in terms of the amount of variation and what has been produced over just 30 years, realistically, 35 years.
Yeah, it's very impressive. It's kind of like that thing.
There were, sorry to interrupt. And again, I got my first hognose snake 30 years ago, or bred mine the first time 30 years ago. There weren't albinos. There was no leucistics in captivity. There was no, you know, there was very, very little in the ways of genetic variation. And in fact, there was a friend of mine in Belfast that produced the first, I think, jaguar hognose snakes, whatever that trait is.
You still see them around. Yeah, that's like, it's from the UK, and then there was
It came out of Belfast. It was a friend of mine that I knew. I would sell boas, and he bred hognose snakes. He only had a pair, and it was the first time he bred them. He produced these. I think it's a pattern variation. Is that right?
Yeah, it's an aberrant pattern. Yeah, they kind of have like these big I'll look up a picture and show you. A lot of times they're lighter gray too. I mean, they selectively line bred them where it wasn't really about the color though, but as your line breeds something with pattern that's aberrant, generally some pigment change comes along with it.
Yeah, that happened. They popped up in his first clutch that he produced. It's very likely the animals that he used were from the hognose snakes that I produced.
Oh, Ireland, really?
Earlier, because I produced a whole bunch of them in Northern Ireland. Yeah.
All right, here's one. And see, they got kind of like broken up aberrant pattern. Oh, this picture is terrible. Sorry. Okay, there's one picture. Is like this one's like glowing. Now they don't all have this color. This is a very pretty one.
Oh wow, yeah.
But yeah, sometimes they're like kind of like see where the neck where it's like you got that kind of twin spotting, but it's like irregular. It's not consistent. Like this one has a more kind of consistent. I mean, they vary. I mean, that's a really pretty one. Right. And then here's like kind of like a lower expression one.
Yeah, that's pretty cool looking. Yeah.
Yeah, they look good. I love Polygenics. Yeah. And this is like what I would consider like kind of like one of the ones that you see a little more often where the pattern's a little more askew.
Right, okay. Yeah, I see that. Yeah,
that's a lot of the really good Jaguars have like really askew pattern. Although all those ones I showed you were like really good ones. And then this is another one, the P-type Jag. And this is an albino. And here's the thing, it looks like an anaconda, but there's no anaconda in it.
Oh, that's neat. Yeah, that's cool.
Oh, and Dave's back. I gotta add him to the stage.
Here's one question I gotta ask you about hognose.
Sure.
Have you seen before where they have enlarged hearts?
Yes.
I've seen my first albinos that I bought, all within 6 months, all pretty much their hearts just blew up and then they died.
Oh, weird.
For a period of time.
I mean, I don't get it too often, but one of my Swiss chocolates got an enlarged heart and died. I had a toffee belly that I bred, and she was like 4 or 5 years old. She's about 4. And she was doing really good. And then out of brumation, she had a big mass around her heart. And then she like died very shortly after. Yeah. And then I've seen some people where they had a hognose die with a what appeared to be an enlarged heart. And then the necropsy shows there's a lot of like fat formed like around the heart. And the heart wasn't large, but then there's also like this unhealthy, irregular development of fat that occurs around like their heart sometimes. So I think that's like I supplement a lot. I try to vary their diet, and I'll fast my hognose sometimes. And so I'm not really like, I mean, I have a lot. I have like probably more hognose than like almost like in the United States, probably than anybody else. You know, I have a lot. I have well over 1,000. And I don't really see I mean, it happens. Like I just had one pass away from it, but I feel like it's maybe like once a year out of like a couple thousand, at most. Right. But yeah, I did one time. Oh, this is another one I had when I was like 17 years old. I had like 2 females back to back that were 2 years old, getting close to breeding size, all within a couple weeks of each other, die of lymphoma.
Oh, no.
Yeah. And because I got necropsies on them, and my vet was like, "This I have lymphoma." I had one die like it was like a few weeks later. same clutch, lymphoma. And then about a year later, I had they were New Mexico locality ones. And I remember they were actually from this cool locale that Craig Trumbauer had, and I ended up, just having limited ones, and 2 of those died of lymphoma. And they were unrelated, those other ones. And so I don't know if it was like my feeder mice or but then ever since then, it's like, I've never really run into that too much, but every now and then when somebody has, I had this hog that was dying this peculiar manner. I wonder if it's something like that. And there's one person, they had a full necropsy on this hog that died, and it died of symptoms that kind of mimicked a disease, like something like a parasitic disease, like diarrhea, kind of getting a little neurological, which can sometimes be an effect of organ failure. And it was having organ failure, and they had a full necropsy on it, and they sent it to a really good university. And basically the lady that sent it in, she was a vet tech herself, and this snake came back with basically the conclusion was it had enlarged capillaries, and I forget what else. I can't remember. I'd have to look at it, but basically it looked like an autoimmune disease.
Wow. Okay.
There's no sign of a virus. They didn't see. Did you see the cancer? No. And I think the kidney was mostly affected, and liver, and the capillaries were irregular throughout the whole animal. And there's a lot of inflammation. And so they basically, the going conclusion they could come up with was hey, this is an autoimmune response.
Oh, interesting. Yeah, the reason I asked about the heart one is that, and it ties into, you're talking about cancer. You see a lot of carpet pythons succumb to cancer very early.
Okay.
And we know that cancers are heritable. At least the genetic variants that predispose an individual to cancer are heritable. It's only if they get an environmental trigger often that will cause that gene to upregulate and to become cancerous. So I was curious whether there was something like that in hognose snakes, and whether the enlarged heart could be something, because I'd heard other people talk about enlarged hearts in hognose snakes, and I had that one time, and that would have been very close to whenever I moved to the US. So maybe I moved to the US 20 years ago. So maybe this was about 17 years ago, because I was living in Raleigh, North Carolina at the time, and I bought some from Frank. What's his name? Frank Patinella. Is that it? Do you remember that guy, David?
Yeah, I remember Frank. Worked for Rich for years. Cop from Long Island, or not Long Island, New York.
Yeah, cold-blooded something or whatever he was. Yeah, I picked up some from him, and I got them home, and they were great. And then their hearts started swelling up, and I was like, "What the shit?" I never necropsied them, but they all died. Their heart just, for young animals, it looked like they'd swallowed a ping pong ball at the end.
Yeah. That's what my female passed away. It looked like she had a ping pong ball, in her, in her throat area, or right where her heart's at, a few inches down her neck.
Yeah, crazy. Dave, where are you going? It looks like you got your bed in the back of the car.
Oh, I've got I'm doing the Nashville Show Me a Reptile show this weekend.
No way. All right, I'm going to be in Knoxville.
Okay. What is that, a Repticon?
No, I'm going to my buddy Jonathan. You've met my buddy Jonathan. He lives in Knoxville.
Yeah. Okay. Tight.
We're going to go down and hang out for a couple of days. If I hadn't thought about it, we probably could. So the Nashville one is this weekend?
Yeah, it's going to be this weekend. It's a new facility. It's something that looks a little bit nicer. It's called the Gaylord.
Yeah, that's a big fancy hotel chain. There's one of those in Maryland.
Okay.
And
is it hot?
I was there a couple of years ago at a meeting. It's a fancy hotel chain. It's got conference center and hotel stuff built into it. Yeah. So sorry, this is the Show Me one, or is this the All American one?
This is the Show Me. The All American is going to come back up late October, I believe, and it's going to be in the same place as the the fairgrounds, the All Pet Expo. This place is northeast Nashville, but the facility's supposed to be really nice. I think it's near a really large mall, but we will see. This is going to be something a little different. I like Nashville, but to be honest with you, the shows have been getting worse.
Yeah. Okay. Yeah, I know there's the All American one at Fredericksburg, Virginia this weekend. And if I hadn't have been going down to Knoxville, I probably would have driven over. It's about 4 hours away. But I want to. I vended that Atlanta show, and it was the first time I ever vended a show. And I want to do another one or two. I'd like to do maybe 2 or 3 a year. And I'm just trying to find out ones that are relatively close to me. It'd be nice to know what the Fredericksburg one goes like. They collapsed. They're not doing the Charlotte one anymore. So it'll be
Yeah,
interesting to know that.
I'll get some reports back from that show. I've got a couple of BOA guys I know that'll be there. Chad will not be there this weekend.
Wow.
Steve Beamer's going there from Nashville, which I thought was kind of funny seeing there's a show in Nashville this weekend. But I got some pretty sensible guys up there. I'll be able to give you a pretty good idea what that show does this first time around.
Yeah, cool. Yeah, good to know. So how long's the drive for you?
Yeah, it's like 7 hours. It's my closer shows. I don't do a lot of Missouri shows. Literally, if I want to go to a show, it's probably going to be a 6-hour drive.
Yeah, okay. Very cool.
Yeah, not too bad. Not too bad. I feel like I sidetracked the conversation. We were doing genetic talk, and now we just talked about bullshit shows. I've lost all train of thought.
Sorry, I apologize for that.
No, it's okay.
That's not your fault.
No, it's good break. We've talked quite a bit. There was something I was gonna bring up. I think definitely. I just gotta think of it again.
Well, Junior, how about this? Are you going to be doing the Scottsdale show for the plant and what is that? The All American Plant and Reptile Expo?
Oh, in Tucson, yeah, I will be there.
Is that Tucson or Scottsdale? Because I thought the very first weekend of November was a Scottsdale show. You might be talking about the other.
I'm in
show.
the very end of September. I'm in Tucson.
Okay, interesting. I think that weekend there's a Kansas City show I was going to do locally. I've been trying to go to Arizona for years for a show. I've never had the opportunity.
We're supposed to go herping. I'm staying an extra day and a half to herp for Mexican hognose.
Okay, well, that's awesome, man. Who you going with?
My friends. So you're included if you want to go.
If I show up to Scottsdale?
I'm not gonna. You're invited too, Warren, but I don't know if you're going to Tucson in the end of September.
I'm not.
Yeah, I want to. When I'm in Scottsdale early November, I looked at the temperatures. It's probably going to be a little too cool. I think the day highs are in the 70s and the nights are in the 50s on average. I don't know if there's going to be that many active reptiles in Arizona around that time.
When we're going, it's actually a pretty good time. We're going to very end of September early, so we're right in time for Hogtober. It's going to be very beginning of October. It's the weekend before Tinley, though. Let me see.
Should be 2 weekends before Tinley.
Is it good? I don't know, dude. That's a far drive. That's like a 19 or 18-hour drive from where you're at. I think it's like an 18-hour drive, Tucson.
Yeah, so I think similar to you, I'll probably start doing the fly to shows. I think I sent you that one deal. What was that? Frontier, like $299 for unlimited flights.
Oh wow.
Yeah, that'd be good. Yeah, fly out there.
Check it out.
I'm going with Joe Kovacs going with me. He's driving. His parents live there, so they're driving me up to Tucson. And I'm flying in, of course.
Oh, that's awesome, man. I love that. So Warren, what is your schedule? When do we actually start?
You just broke up there, Dave.
That was a rough question.
I can make anything up to answer that. Try it again, Dave. What was your question?
Oh. It was an awful question. I was just going to ask you, are we going to see you at Tinley? Are there any of the bigger shows that you'll be venturing to this year?
I'm not this year. I'm planning to go to Tinley in March because we have a USARK board meeting, and I've been on the USARK board since it became USARK. So I plan to fly in for that. I don't plan to. I talked to Bob Ashley, and he said if I wanted a table, he could probably make that happen. But I don't think I'll have enough to bring up there to make it worthwhile driving instead of just flying over. So but I'd like to go to Tinley in March, and then I want to vend. I think if I've got enough left, I'm going to vend the Atlanta show again in January, end of January. I'm curious what people are going to think about the show being in like January 31st or whatever. Is that too close to Christmas and not close enough to tax season, or is it? How do people think that that's going to go?
I think that's a good time. I think that's a good time to have show.
Yeah.
Yeah, I don't think it's a horrible time. You know, there's a lot of people that are doing their taxes like immediately this year or every year. I find tax money is actually hitting the shows in like February and March compared to we're used to think it'd be like April. So I honestly don't think. I don't know. Like you said, the rule of thumb doesn't play like it used to. Like I used to do really well through like April and May. This past year, I thought my May slowed down a little more than my April did. So tax money didn't last as long as it normally does.
Yeah, so I wonder, is it, is Christmas money going to be the driver for that end of January? Because tax money is not going to be in by the end of January for people. So it's
true.
Yeah. But I did well.
It really needs the buy now pay later feature.
I did well in Atlanta.
What was that?
I did well in Atlanta whenever it was there, whatever, in August. So it'd be nice to do that one again. I think that's going to be
January should be better.
You think so? Yeah.
Yeah.
I'll definitely do it.
Summer months are tough, man.
Yeah, summer months are kind of tough. I mean, sometimes there's some exceptions with like some really good expos because it can bring out the people.
Yeah.
But expos in the winter and spring are like definitely, I'd say, better on average than ones during the summer. And the thing is too, like expos, kind of, in my opinion, and from what I've seen, like the bigger ones, they definitely bring in those people that are waiting for that big particular expo because it's going to have the bigger, better breeders and people they want to see, and like they're more fun, and so it's kind of like more of an event versus like a monthly show. But there's so many expos now. Some people like pick and choose, and I think like even like for like Tinley, usually March is better than my October.
Right.
And I think Southeast is going to be the same. I think January is going to be a little better.
Cool. Yeah, I'll sign up. I'll do it again. It was fun. It was fun the last time.
Yeah, that's a good show. I like that one a lot. It was a lot of fun.
Yeah, it was bright. Yeah. It didn't feel like it was just maybe the snake discovery thing made it a bit difficult, with so many kids running around, blocking the doors and all that there. But I think everything else seemed to go really well from that.
No, but I will say I thought they navigated that crowd really well. They didn't bring them through the main door. They kind of brought them through the side door. I didn't think it was that congested because Snake Discovery being there compared to what I've seen them at other shows where it's literally fucked up the entire show.
Wow. Yeah, it's not good. Yeah. Well, I'll do it again.
So what do you say we do a little, you guys want to make a wrap-up conversation, maybe talking a little USARK? Warren, you probably have more of an idea of what's going on around us than anybody else.
I should do, but I don't, because I've been so invested in my other work lately. So I need to sit down. I think we're going to be having a meeting soon, and we'll kind of get all of the updates. But so far, I think we've shown that USARK has just been very, very successful. With Phil as our president, he's done really well. With having people like Vanessa that really helped out at the shows and drives a lot of that stuff, it's done really well. So I'm pretty excited about the future of USARK. The big thing is that people need to remember that, while things are being won, the other things will come up, and it costs a lot of money to pay for retainers on lawyers and to pay for, retainers on various aspects. So, running USARK is not inexpensive. So keep up your membership, keep up your donations, and keep up supporting USARK.
Definitely.
Yeah, I think the membership thing, buy merch. The membership thing is such a tough thing with that. I bring this up every time we have the conversation, but I talked to Goss, and this again was like 2020, and there was a surprisingly low amount of people actually getting a membership. And I remember where we talked about like back in the day, Jeff Ronnie's website, Ralph Davis's website, like on the front page, you'd see gold member, USARK. And I really think we need to bring that back, make that trend where like your Morph Market account always says gold member, silver member, which it might do already. I think there should be like a watermark that you get for your Facebook business page. You could put on your logo if you want, saying that you're a member of some kind. Because again, memberships are really low, and I'm sure you know that.
Yeah, but the good thing with memberships you can do now is we've got one where you can pay it monthly. And it's like I think it's $5 a month or $7 a month. You know what? People are going to go and spend over $5 getting a coffee at Starbucks. You know,
Yeah.
so it costs nothing to put one of those on those rolling kind of memberships, and they're a great way to donate, but not to feel like you're sending $70 off to somewhere or $100. You know, it's a great way to donate and to not feel the effect of it. You know, and also if you're selling animals, another great way to do that is to say that 10% of your sale goes to USARK. You know, or 5% of your sale, or something. You know, and try and do that kind of donation here and there.
No,
Yeah.
I agree with that. I think what Reptile Basics, and I'm sure they still do it, they had the roundup feature, I want to say, where you can make a donation to USARK for years. I think a lot of the other shipping companies offered that for a long time. I think there's a lot of little things you can do to boost it up a little bit, especially in the shipping department, man. You know, saying I just want to round it up 45 cents, a lot of people round that up 45 cents.
Absolutely. Yeah.
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 That's something that Darian should apply to the website, because I know Ship Your Reptiles had that and stuff too, where it's like, you know, you can donate, you can round up or donate $1. Because if you start getting like everybody to do that per shipment, or maybe Darian could even implement something and force people. I don't know if people would like that, but like, I don't know, like $1, just $1. And so for every shipment, like $1 goes to USARK, man. You know how much money they would raise? I think they had like 1 week, 10,000 shipments. They'd be raising like 40, 50 grand a month.
Yeah, it's amazing.
So that's how you guys can help. It's like other breeders you're listening. Yeah, just like donate like I don't know, keep a mental note if they're not going to have that feature. For every like few shipments, just send a couple bucks. If you send like a few dollars, like that's what I used to do is Ship Your Reptiles. You know, I do usually a hognose auction a year. You know, donate stuff at the auctions, and then you know, I have my membership. You know, but I mean, I have a little bit of disposable income. I'm not saying everybody should do this. You know, some people, you know, especially when it's starting out in reptiles. I remember when I was, you know, my dad and I would go to these shows, and you know, I was really wanting to pursue this as like, you know, not only a hobby but to make money. It was not that easy. It was not that easy at all. You know, you go to a big expo and you know, took all this time breeding stuff, and you know, it made like $2,000. You know, and then after having to pay for everything, and my dad's like, "Oh yeah, some of that money has to go back to this project that we invest." I'd have like $150. And so, anyway, you know, things are expensive, but yeah, that's like, so if everybody's like shipping just donated $1, that would be more better than just like some people here and there donating $70, because like that dollars would accumulate across these thousands of like people. Because Darian said like when they were first like doing a morph market shipping, like the first few months, like the 1 month they had like 10,000 shipments, and I know more people are using them now. So
yeah, like I said, the dollar donation, or again, even just the round up. I mean, you've gone to fast food places, you get your order, and they say you want to round up 45 cents to this foundation. A lot of people don't hesitate on that. You know, 45 cents is less offending than $1 for some people.
exactly. Yep.
Yeah, but no, I think there's always a lot of different ways that, you know, we could find ways to raise money. But, you know, I don't know. I don't even think we're allowed to talk about it, but do you even know how much it costs per year for USARK to function, or that's like a no answer?
I do know it. I don't know it offhand.
Tell us off. Tell us behind the scenes.
It's a massive amount of money. It's a massive.
And
they need more. They probably need more anyway.
Lawyers and for lobbyists are very high. You know, so it does cost a lot of money. You know, and then you just think about it. You know, it costs money to attend every meeting, every show that Phil goes to, or Vanessa or whatever. That costs money to get there. It costs money to have everything printed. It costs money to produce all the T-shirts and so on. They don't make a lot of money on those kind of things, but it costs a lot of money to get them done. So to run it is super expensive. So therefore, every year, just like any business, you got to knock off all of that stuff first before it's becoming profitable. And where it's profitable it's not profitable money going to the board of directors don't get paid. Right, so I've been doing it for 17 years. You don't get paid for it. Phil gets a salary. and Vanessa will get a partial salary from it for the amount of work she does. But yeah, all the rest of that money, it's a non-profit essentially. So therefore, that money is going in to fight, these bills and these issues that are popping up all over the place. Just go to the website, you'll see this county and that county and this state and so on, all have popping up all of the time.
Yeah. So I bring this up all the time, and Warren, you probably have heard me talk about this, but I always make the comparison. One of my dad's ex-wives worked for the Boys and Girls Club. Her job was to raise I think it was either $3 or $5 million a year to keep it going. And her salary was pretty much based on that. She was essentially making commissions off of bringing in money to run the Boys and Girls Club, which I don't really see a problem with. Has there ever been any talk within USARK where they have a sales guy come in completely based on commissions? The guy that can go sit down with Petco, go sit down with PetSmart, really get out there and try to find these big donations to come in.
Yeah, I don't think so. I think the people that are on the board, specifically certain members of the board, have a lot of those contacts to go towards that. And Phil has got such a good reputation within the industry that I think he's the guy that actually is able to make that stuff happen.
That's fair. And as far as you know, has Petco or PetSmart ever donated to USARK?
I am not aware. Okay. ZooMed definitely has. ZooMed is a very large sponsor for USARK. Without ZooMed, it would be a very different scenario.
Understandable. Junior, what do you got, buddy?
I think that's about it. I was going to ask Warren one other quick question. I'll just phrase it like this: with the sequencing of the genome to compare, let's say they're trying to find common alleles, or what you're we were talking about earlier with finding how closely related animals are. Some people were talking about, and I don't think it's possible. Basically, do a DNA sequencing on animals that, say, have polygenic traits and trying to identify polygenic traits. I'm like, you're not going to be able to do that. That's going to be I think almost nearly impossible. Depending on but if there's a culmination of a lot of alleles contributing to a trait, that's going to be very difficult to sequence. And because any of those animals that are related to that line are going to have so many common alleles, you're not going to know which ones are doing what.
That's exactly right. But polygenics makes things a lot harder to identify, because it could be 2 genes that are interacting, or it could be 20 genes that are interacting. And what you don't know in each individual is which combination of those genes do they have, which allelic variants do they have of those, and therefore it could be really difficult. You might be able to turn around and say, if we compared 100 animals, we can definitely cluster animals and say these are showing a higher propensity of shared alleles than these here. But being able to knock that could be, that could be 20,000 alleles that are shared.
Yeah, right? Okay.
It's different whenever it's a single recessive trait, or even an dominant or a dominant, where it's probably a single genetic variant in a gene. Remember, genes are thousands and thousands of base pairs long. So we're hoping to find that variant. And we need sufficient numbers of individuals that have that variant and don't have that variant to be able to identify that variant. And it takes time. If you've got any related individuals in that set, then you've got a higher proportion of alleles that are shared just by chance, and they're not necessarily that trait that you're looking for. So therefore, whenever there are new traits, you're going to have to eliminate the inbreeding effect and the close relative effect. But with polygenics, it's going to be a lot more difficult to actually be able to turn around and say, yeah, these are the 10 gene or 10 variants that are associated with this trait.
Okay.
It's just because you don't know, you can't turn around with an individual and say, this is carrying all of the variants that make up this polygenic trait, whereas this one's carrying 1, and this one's carrying 3, and this one's carrying 5, and that kind of thing. You can't pigeonhole them and say, it's here, it's here, it's here, right?
Right. And then since a lot of the polygenics, there's a degree of inbreeding, so then there's going to be all these other common alleles that have nothing to do with the ones you're looking for, and you're not going to be able to decipher them.
That's right. So we can narrow it down to the subsets of genes, but that could be thousands, it could be hundreds, So it's certainly at this moment in time, it's not as easy. In time, it will likely become easier when more individuals are sequenced. But remember, whenever RGI or anybody are doing the shed test, they're not sequencing the genome; they're sequencing a variant. So they're not getting the variants in other genes unless they're not getting variants across the genome. So you can't really use those data to build up other stories down the line. It's just doing a PCR test ultimately to say you have it or you don't have it. Whereas to get to that, you need to sequence dozens or hundreds of individuals to be able to identify those genes. And when it comes to polygenics, you're into even greater numbers than that there.
Yeah, thanks for the explanation.
So I have a question. Well, we may have 10 more questions, but I got another question based on what we were just talking about. I had a conversation at the Atlanta show with a ball python breeder that made a comment that he's going to start testing every baby shed before he sells it because he was worried about partho babies when he's starting to make his double, triple, and quad hets. Even if you make a, let's say, single partho baby in a clutch, that female is more than likely not going to reproduce. So testing a shed really doesn't matter, or does it seem like not a waste of time, but does it seem like a little extra?
Partho babies, the problem is that they often don't make it to adulthood. They very rarely make it to adulthood. They depending on whether they're a colubrid or colubroid or whether they're like a boa or a python, they just don't do well at all. I've got about 140 parthenogens, and they're all but 2 of them are living in my freezer at work. And they range from a day old to generally about 2 years old. I've only ever been able to raise up 2 parthenogens to adulthood, and that was one boa and one ball python. And I got them both to reproduce, and then they both didn't recover from that there. So it's parthenogens are not something you want in your breeding program. You probably want to be able to identify them. If there's a fear that you're selling that a clutch has parthenogens in it, then you'd want to identify those and not sell them, because the person that buys them are not going to be very happy whenever the animal dies very young. And that's just not a trait with snakes. It's the same trait. It's the same with other species that produce parthenogenetically, facultatively parthenogenetically. So birds, the elasmobranchs, which are like sharks and rays and so on, crocodiles, you'll see that the parthenogenetic offspring don't survive generally to adulthood to reproduce.
Has it been shown in any species where parthos do okay, other than like the Mediterranean house geckos?
Yeah, but that's a different form of parthenogenesis. That's an obligate form of parthenogenesis, and the reason they do well is because the offspring retain the genetic diversity of their mother. They pass down unreduced eggs, so they're the complete complement of genetic diversity of the mother, where it's a facultative parthenogen, ball python or a boa, a crocodile or a bird. They have an egg which fuses to a byproduct of that egg's production called the second polar body, and that second polar body contains a set of chromosomes that are almost identical to the egg, but not completely identical. So there's a small amount of genetic variation, but that variation is in regions of the genome where there's not a lot of genes. So those offspring that are parthenogens are essentially as inbred as you can get them in a really bad way. They're homozygous across most of their genome. So they're not good. Whereas in a Mediterranean gecko or house gecko or whatever, Moorish gecko, they are genetically diverse.
Okay. I hit another partho ball python. It was a single female and a clutch. Well, it was a clutch of like 7, but I bred like a female super blackhead pastel lesser to a blackhead cinnamon and made one white snake. Everything else in the clutch made sense except for that one, of course. Yeah. So a single partho, but of course, the same way with boas, we started noticing parthos once we started having more genetic diversity and we knew what our odds were supposed to be.
Mm-hmm.
I'm wondering, I'm sure it's been very common in ball pythons over the years. It just hasn't been as noticed as other species.
Right. Oh, absolutely. It's just because we've got so much genetic variation and we understand the genetic traits, we understand you bring to that, you get this. And whenever we don't get this, then we kind of know where to start looking. Yeah.
Junior?
Oh, I was gonna say, well, you said that guy wanted to test all of his ball pythons for parthos?
Well, what he wanted to do is every baby he produced, he was going to have the females shed checked to have essentially genetics to go with the animal. But he said that based on partho clutches or the possibility of partho clutches.
Okay. Well, you can also learn what genetics the ball python has, but from what I know, and I don't know anything really about parthogenesis, but it's not super common with ball pythons.
Yes and no. I know of 3 different clutches in the last month from just 2 different breeders that we ran into parthenogenics.
Really? Okay.
Bob Vu, I had an animal over there for a new project he's working on. He bred it, and the female parthoed. So he ended up making a bunch of clone copies of mom. There wasn't any dominant, so it doesn't look like there's going to be a super. But right away he saw that. Like I said, I had 1 leucistic in my clutch, but I will say, the industry ran into like a bad run of like het sunsets for years there, where everybody was buying het sunsets and none of them were proven out. And there was some fuckery there, of course. But back in the day, everything would come in with a certificate. Ralph Davis would draw a certain part of the pattern like on the actual receipt, so we can match up the animal. It might not necessarily be a bad thing for ball pythons to start testing sheds on females before selling it. So at least there is some kind of paperwork again, because I said, it stopped being the norm. Like, I've been buying hets for the last 10 years. I never get any special paperwork with my hets anymore.
Yeah, just remember that when testing them, it's only if they test them and they say, right, I bred a homozygous to a wild type, or to an individual who was wild type with that gene, they're expecting all of the offspring to be heterozygous. That's the only way that those shed tests will be able to identify parthenogens and so on, because instead of so to identify parthenogens, you're actually better doing screening either whole genome or partial genome sequencing, which becomes expensive to do it realistically. That's why I don't test parthenogens for people anymore. It's too expensive. It's the only time I'll do it is if it's a species we haven't identified it in before. In case androgens are what we're really interested in. So Justin sent me up he sent me up a couple of parthenogens, and he sent me up that androgen puzzle, whatever, that he produced earlier this year. That androgen died just a couple of weeks ago, and the parthenogens seem to be doing fine at the moment, but they'll keel over at some point in the next 2 years. But it's expensive to actually test to prove parthenogenesis now, and your shed testing is going to only do it if you're expecting to find heterozygous individuals that are actually then homozygous wild type or mutant.
Here's one other question, because this is a conversation we've had a lot in the past, and now I know Charlie and Benjamin are starting to work on it, but shed testing for sex with northern blue-tongued skinks, or any blue-tongued skink really. There was a paper I haven't read yet, but it kind of broke. I heard there was some fishery about the paper. Like, it didn't really make sense, but it made sense. What has been the biggest hang-up with being able to test for sex in these zones?
Depends on the species. With green tree pythons and with Morelia, they got a really screwed up genome. So I'm involved with a group right now where we're sequencing the carpet python genome, and hopefully that will give us a better idea of being able to identify sex chromosomes, and therefore from that we'll be able to work through green tree pythons and carpet pythons and so on. With blue-tongued skinks, it's impetus to do it. I was approached a number of years ago by a well-known individual. I think I've told you this story, Dave. I'm not going to elaborate who it was, but I was approached a number of years ago by somebody that got into blue tongues in a big way, and approached me because we just published papers on identifying sex chromosomes in boas and pythons, where we identified the XX, XY sex chromosomes instead of sex. Whereas before that, we thought that boas and pythons were ZZ, ZW sex chromosomes. This individual contacted me and asked me would I be interested in developing sex determination mechanism or methods for blue-tongued skinks, because they believed it was going to be the next big thing in the hobby. And I said, yeah, I'm interested in it. And when I told them the price to do it, they said, well, if you fund it, I'll help you market the sales. But they wanted to take 70% of the revenue of the sale for each test because they were marketing on it. They were going to market it on their website, of which I told them to kindly go themselves. And I didn't need them for that there. We could develop a test if we wanted and market it ourselves. I'll tell you who that was next time I see you, Dave, or Jeff. But
Okay.
it was not somebody you might expect it to be, and therefore I don't have a lot of respect for them. But yeah, it's impetus. It's somebody would need to invest the money. It takes time. It takes money, and we also have to have known sex individuals, right? So we just need it. That's easy enough to get from people that have larger collections. But realize that it's not just the money for the test; it's the money for the person to do that. So if I get one of my postdocs to do that work in my lab, I'm paying them, $66,000 a year plus fringe. So in university, that means that I'm paying them combined about altogether their salaries costing me about $90,000. That's to do work related to projects that we're involved with my lab. If I then take it out of that and say, right, for the next month you're going to be working on, blue-tongued skink sex determination because this person's interested in it, that's a lot of money to invest to develop that test, not just for salary, but then for all of the consumables and the sequencing and so on. So you could be looking at 20,000 or more to develop the test. And then you're hoping that there's going to be enough revenue coming in of people saying, I want to get my blue-tongued skink sexed. So the ball python stuff has worked really, really well because ball pythons are the biggest animal in the hobby. And I would imagine hognose snakes might go as well. The only problem with hognose snakes is the large clutch sizes. So therefore it makes it more expensive to do all those shed testing depending on what you want to do. But there's I think the whole trajectory that people thought was happening with blue-tongued skinks didn't happen. People thought these are going to be the next, bearded dragons and everyone's going to have them. And I don't necessarily think that happened. So I wouldn't personally invest the money to do blue-tongued skink sex determination because I think it would take me a long time to recoup the revenue from that there and then start making a profit from it.
So
whereas green tree pythons, however, I think I could probably do that and make more money from it because of the demand for that there.
No, and I understand that. And the one thing is in this hobby is there's always the next big thing, and we always talk about it, and then very rarely does it happen. Do I think that blue-tongued skinks are a great alternative to a bearded dragon? Yes, but they don't keep up with clutch size. The jactiver, they don't have as many different projects. Not that bearded dragons have that many different projects. It's a lot of line bred stuff similar to what we're doing in blue tongues. And it will be interesting to see with morphs being readily available in the United States now where that market's going to go. Because up until honestly this year and last year, it was very rare to have an opportunity to buy a mutation. And I think the only thing that I keep on saying kind of hurts us now is the community's small. We now have 4 to 5 to 6 different mutations you can buy. And I want to say they're all $10,000 minus one of them. And there's just not that kind of money in that community. So it is going to take people from other communities that enjoy the species to move into it. And that will increase the value, of course, as the demand gets higher. But right now, there is a little bit of a lack of demand.
Yeah, so bearded dragons have done so well with a lack of genetic morphs. You get the high reds and high yellows and so on, but 99% of the kids that own bearded dragons own a bearded dragon, a wild-type bearded dragon, basically. And the morphs are basically being swapped among the breeders, and then they trickle down. Think about 20 years ago, 99% of the kids that had a ball python had a wild-type ball python. And whereas now 99% of the kids that got a ball python have got some kind of morph because the cost has trickled down so much that they can now get them for peanuts, right? So it's a whole kind of demand issue. So blue-tongued skinks, give it another 10 years and you're going to see a lot of kids that have blue-tongued skinks that have got an albino or a melanistic or whatever, an exanthic or whatever. So I think you'll see that, but it just takes time in this hobby.
Well, when Bill Brandt started buying these, I think it was probably it might have been as far as 6, 7, 8 years ago. That was kind of his idea. These could be in every Petco. But I think after a couple of years of breeding, there was just an inconsistency. Yeah. And because of that, it didn't seem like something that could be produced in high enough numbers to put in, again, like every Petco in the United States.
Yeah, think about how many eggs a bearded dragon produces a year. Like even leopard geckos. I think I told you, Jeff, my son got into breeding leopard geckos this year. He's 7 years old. He turns 8 next week. I bought him a 1.3 group of pretty expensive leopard geckos. 2 of his females produced. He produced 19 babies this year.
Yeah, that's fast.
Geckos.
Yeah, crested geckos, leopard geckos, bearded dragons, they're like among of the, probably top 3 more prolific, commonly kept species of reptile. And that's the thing, leopard geckos, bearded dragons, and even crested geckos. If you have a large enough collection, you can start to supply, a lot of shops and, that demand.
Yeah, so whereas blue tongue skinks, what is their average or litter size? Is it like 8 to 12?
8 to 12 is exactly what I was going to say.
And then are they producing yearly or is it every other year?
You know what? I had one female that was one of the early founders for my collection, and honestly, I want to say that she gave me a litter the first 7 years I bred her, and then since then it's been a little more sporadic.
Right. That's pretty incredible still. But you look at a bearded dragon and you're going to get multiple clutches of 20-something eggs. So there's 60 or 80 babies versus 8 to 12. So they're a totally different scale for how they can be marketed.
I think skinks will always have a spot, but like I said, the community needs to grow. If anything has shrank over the last 7 years after exploding about 7 years ago. It's just going to take people over the industry to show interest in the species. The fact that you can keep them in rack systems, I know is an incentive for a lot of people. But no, I had known a lot of guys, like even we brought up Steve Sykes earlier. Steve went pretty heavy into blue tongues for a minute there, but I think it was around that same time we weren't allowed to export anymore, and I think that's when he got out of the species.
Right. Okay.
Yeah.
Hey, I got a quick question for you, Jeff. Do you keep hardwicki? Do you breed them?
No, I had them for a little bit, not too long. I bred them like 1 year, and then you'll like this one. I bred some hardwicki to some bold striped leopard geckos. I wanted to see if you could hybridize them. Never got any fertile eggs though, but I don't have any hardwicki. I only had them for a short bit. My favorite of all the Ubiloferis would be the Angramanu, though. I like the Iranian leopard geckos.
They're kind of big, long, and skinny.
Yeah, they're like basketball players of the leopard geckos. They got like even their hands are big, like their toes are long, their hands are big. They're real lanky. They got big heads. They're the biggest natural species of Ubiloferis. Maybe aside from the giant overfed Macularius, those might be a little bigger mass size, but not including those, Angramanu are the biggest.
Yeah, I like the hardwicki.
They're cool. They look like a in between of like a leopard gecko, a cave gecko, and like an African fat tail.
Yeah. I do like those. I keep thinking about getting some for my office at work.
Yeah, they're neat. I like them. I only have Macularis. I don't even have Angramanu. Angramanu are my favorite. I only have those. I have Macularis, and then I have African fat tail geckos. Hemidiconics.
Yeah, you need to do a show on leopard geckos. I've become, after getting my son into them, they were the first thing I was into. I've got massively into them again. So
they're fun. There's a lot of stuff you can do with them. Sometimes the maintenance is a bit much. I'm a bit burnt out. I'm pretty much all hog nose, but I still do the leopard geckos. I still like them, but
Yeah. Do a show.
Hey, Junior. Oh, go ahead. I'm sorry, Warren.
No, I'm just saying you should do a show on leopard geckos. We could do it like Steve Sykes on or someone like that there.
Yeah, I should actually. Yeah, I haven't done a leopard gecko show. Maybe I'll have a leopard gecko person on. I never really thought of that. It's a good idea, actually. That is a good idea.
It's time to get that going again in the shows, because I was amazed walking around the Atlanta show and seeing how few leopard geckos there were at the show. I thought there'd be a lot more.
Well, that's not common. Usually, most of these shows there's too many. Atlanta, there was wasn't that many there.
Okay, yeah.
Hey, Junior, when you work with fat tails, were you ever successful feeding them mealworms over crickets, or is crickets a staple for their diet?
Crickets are a little better. It's a leaner diet. You know, these larval form of insect feeders are a little higher fat. You know, like mealworms, waxworms, silkworms. You can get the African fantail geckos to eat the mealworms. A lot of times you got to kind of like convince them and train them to eat them. And once they realize that they're prey and they get them to eat them, they stay on them. But the fat tails really like to hunt. They really like the movement. That really stimulates their, you know, desire to, you know, eat. They really identify prey through the movement. So when you give them a mealworm, sometimes they're just kind of like crawling slowly in a straight line to like look at it, and they're like kind of like disinterested. Sometimes they don't give a shit about it. They look at it and then they kind of lose interest. But once you get one, like you break one open and you put it in a crook in their mouth and you kind of like rub it back and forth, and you let them lick it and taste it, and you get them to eat it, like either hand fed or tongue fed, then usually they'll start taking mealworms after you do that a few times. But yeah, a lot of fat tails, if you just throw in mealworms, they look at them like, what the hell is that? And then they like lose interest.
What are you mainly feeding them?
Crickets. They'll do dubia roaches. Crickets are actually more prone to eat superworms because superworms are a little more active than your basic mealworm. So a lot of it's just really the movement.
And for your leopards, you're mainly mealworms?
Yeah, I do mealworms. Like, well, we just started. Actually, my girlfriend Crystal is her idea because I was like, you know, the crickets are a bit messy, and you know, I do think crickets definitely are a healthier staple diet over mealworms. The mealworms are a bit fatty, but mealworms are a little easier. So we are breeding mealworms. But yeah, the fat tails, I really like to do crickets. You can do dubia roaches too, or you just vary. You know, you give them a little bit of everything. Now, leopard geckos, I've heard of people like saying like, oh, I can't get this leopard gecko to eat like mealworms or whatever. I don't think they gave them enough time. I never had a leopard gecko that wouldn't eat both crickets and mealworms.
Yeah, ours do just mealworms, but it's never offer crickets. But it's it was interesting. We got dubia roaches for them, and they're less interested in the dubia roaches than the mealworms.
It seems to be like a learning curve because when you have them like on like a staple diet, and so that's funny that you're seeing the inverse of that because I'd see it more because I did more crickets and I'd switch them to mealworms and it'd sometimes take them a bit. Like when my dad and I had a bunch of leopard geckos, there was this cricket virus, and for a short bit, we were doing all crickets, and all of a sudden we couldn't really get crickets anymore. So we switched them all pretty much to mealworms, and you know, we had hundreds and hundreds of geckos. We never had one that got thin, and we couldn't monitor them, you know, to make sure they were eating because we just had like too many. Like there's over 1,000 geckos actually. Never had one get thin, so I'm sure some of them didn't eat them right away, but they all learned to eat them, and so we never got it. You know, we just started giving them mealworms and mealworm bowls, and it worked.
Yeah.
Now with fat tails, though, I have had ones like, you know, for those we were still able to get enough crickets to where we fed them. The fat tails I never put on a staple diet of mealworms. There was this one girl that used to buy at one point. She'd spend good money on fat tails, and you know, it'd be like not, you know, cheap orders, like a few $1,000s or more. And she'd be like, I'll buy them, but you got to get them on mealworms first. So then I'd have to fucking bust open a mealworm, rub it in her mouth, get them to eat it, pick them up. And then after about a couple weeks, I'd get them all on mealworms. Like, yeah, they're ready to go now. And then I'd ship them to her.
Wow. Funny. All right. Well, that was a tangent, but yeah, just curious because of my they find interest in leopard geckos and hardwickie. Okay.
Yeah, they're cool. Yeah, that's the one problem, Warren. So you gotta be selective with your questions with me. Sometimes I'll start going on tangents. You'll ask me one simple thing, and I'll tell you a 20-minute story how a gecko ate a mealworm.
Yeah, I'm the same because I breed 22 different species of snakes. So
yeah, but it's good. I mean, I like especially talking to somebody like you, and if you can, like, you know, sometimes you hit a core memory or something from it, reminds you of something, and you can go on these like tangents or these, you know, stories, you know, they're good analogies.
Yeah.
Yeah, I think that makes a better podcast when you get longer answers like that, even if it takes us in a weird direction.
Yeah, I agree. Yeah.
So, Warren, what project did you buy your son?
So at whenever we got into them, I traded for some stuff. I got there were Mandarin, Tangerine, Tremper Albino, and I've got it written down somewhere else. Hypo Ghost, Albi Snow, and Ghost. And then off Steve Sykes, I picked up a Tremper Albino, Mandarin, Tangerine, something else. So he's been busy with those. And then at the Atlanta show, I got him some Black Knight geckos. I got one off Jeff. I got one off another person. And my son just loves those. Those things are turning out really dark, and they've only been here for a month.
Yeah, they're cool.
They're a lot of fun. So he wants to breed them into his Tremper albinos. The nice thing about being a geneticist is that my son has got some level of understanding of genetics as a 7-year-old.
That's cool.
Plans to get to this Tremper albino Black Knight and Mandarin Black Knight and stuff. So he's got his plans. Well, yeah, we keep looking, but I think I'm going to try and pick myself up some hard wiki from my office at work. Yeah, that could be fun. Yeah.
All right, Dave.
Now are we done?
All right. Yeah, we've been at 2.5 hours. And actually,
Cool.
I'd love to talk more, but I want to take a nap.
Lucky for some. Yeah, well, anytime you want to chat,
That's
guys,
real.
just give me a shout. I'm always happy to do this.
Yeah, it was your awesome guest, Warren. Definitely like your, knowledge of genetics, so you can ask you questions, you can explain things, and so you're definitely. I like talking to people that have really good knowledge in particular fields, of course, and have information, smart people such as yourself. So thank you for coming on and sharing your knowledge with us.
Anytime. It's always good to talk to good people. I always have fun.
Well, you enjoy Knoxville this weekend, bud.
Yeah, you enjoy Nashville. If I can convince my wife to let me escape, I might drive down to Nashville. You never know. I think it's only 2.5 hours from Knoxville.
Well, I don't know if this is an incentive for you, but I heard you went to the Metallica concert. Nine Inch Nails is playing Saturday night in Nashville. I'm thinking about it.
Yeah, we went to Metallica because we were invited as guests of Metallica. So it's I don't know if I would have paid the money, but we were
I'm inviting you too, but you're gonna have to pay.
Yeah. If I can get Nine Inch Nails to invite me and cover everything, then I'll do that there.
Yeah, it's a tall order, man.
Are you going to a Nine Inch Nails gig?
Am I going where?
Are you going to go to that 9 Inch Nails gig?
I think I've got to go, man. I haven't seen them since the late 90s. I completely obsessed over them in high school. I owned all their Halos. I had 20 of their T-shirts.
I was going to say, I think the first time I met you, you were wearing a 9 Inch Nails T-shirt.
Probably was.
You got to go then.
So weird. There was no stash. It was just a 9 Inch Nails T-shirt, I think.
Yeah, I still hold on to a little bit of that high school wardrobe I used to hold on to, but all that shit's gone now. I brought a very bright pink shirt for the concert.
Excellent. Well, yeah, you have fun. I don't think I'm going to be able to do that far. I think we might be able to go down and do the show and then drive back again. But but if we do, I'll let you know. And if not, just tell me how the show was. I'm curious.
I'll give you feedback, I promise.
Good stuff, man. I appreciate it.
Of course.
All right, thanks, guys. And I'm going to do one quick thing. Reptile Super Show, the Anaheim show, is not this weekend, but it's next weekend. It's like September 12th or 13th or 14th. It's right in that neighborhood. Just look it up. Be there. All right. Forgot to do that at the beginning. All right. Thanks, Warren.
All right, guys.
We'll see you around. Thanks for talking and hanging out.
That's good.
Thanks, as always, to you too, Dave. Appreciate you guys.
All right.
Bye, everybody.
All right. Bye.
Later, guys.